This podcast is independent from my studies at Texas A&M's College of Veterinary Medicine and Biomedical Sciences. This is intended to share knowledge and experiences in the field of veterinary medicine and to share the importance of medicine for the pursuit of science and its positive effects on the general public.
Bummer.
Well, anyways, do you
Okay,
want to introduce yourself?
sure. My name is Virginia Fate, and I'm a clinical professor in the College of Veterinary Medicine and Biomedical Sciences at Texas A&M University. I've been at Texas A&M almost 21 years. I came in as a clinical assistant professor and have been promoted since then. I was hired mainly because I am a veterinary clinical pharmacologist, and so I they needed someone to teach pharmacology or teach in the pharmacology program. And in those days, in the legacy curriculum, there were 2 semesters of pharmacology. So there was a 5 credit course in the fall and a 3 credit course in the spring. So it was a lot of hours. Um, so yeah, that's how I started at Texas A&M.
Okay. Um, and now you're teaching pharmacology. Have you been teaching that entire 21 years
Hmm.
you've been here?
Hmm.
Because you learned pharmacology at Texas A&M from the old professor, is that correct?
No,
Oh, okay.
no. I went to vet school at Auburn.
Okay.
Yep. And then I did a food animal internship at Kansas State. And I also, then I stayed on for a year. I was a clinical instructor. I was on the ambulatory food animal ambulatory service for a year. And then I did a PhD. I went to Iowa State and did a PhD and prepared for clinical pharmacology boards. And my interest had always been in food animal, even in vet school. I thought I wanted to do something related to food animal. And so my PhD was focused on antibiotics in cattle with respiratory disease. I know you're shocked to hear that it was about antibiotics.
I was almost considering just studying antibiotics before this, just to make sure
Oh.
I was like on the nose.
Oh no, you're good. I'm sure you know as much about antibiotics as any second year vet student, so you're going to be great. Yeah, and I have spent a lot of my so after my PhD, I spent about 5 years doing a couple of different part-time jobs. Um, my major professor started or while I was in grad school, I guess we started developing a web-based decision support sort of system that was going to be helpful to food animal veterinarians before we're focusing on cattle and swine, and trying to develop treatment, um, not guidelines, but we did some statistical modeling, pharmacokinetic modeling to take multiple sources of data on about the pharmacokinetics of various antibiotics, and and then sort of match that up with if you had a result from the laboratory with a minimal inhibitory concentration, could you predict what dose would be most effective? Um, and so I did that part time, and I also taught a few courses. I moved to Southern Colorado, Pueblo, Colorado. There's a local university there, CSU Pueblo. It's actually a branch of CSU now. CSU Pueblo, and they had a biology department, and so I went to them and said, "Yeah, I'm in town. How about a?" And they were, they hired adjunct professor is what I was, and I taught. I think I taught physiology, histology, virology, immunology. That was those are the so
To
to
vet
undergrads.
students?
No,
Oh,
no, to
undergrads.
undergrads. Yeah, it was in a mostly an undergrad university. They have a couple of master's degree programs, but it's mostly undergrads. Um, and so I, as an intern at a teaching hospital, you do some clinical teaching. You're with vet students all day, so you might do rounds. You might do some. I didn't do much didactic stuff, but I would definitely have rounds, and um, I was 1 year further advanced than they were, so see one, do one, teach one, and and then in grad school, I did some a little bit of field service, and also did some other sort of a few lectures here and there and didactic kinds of things, and also um, started doing some continuing education kind of presentations, just a few. So I had just enough teaching under my belt to feel like I could say, yeah, I can teach your class. I haven't had immunology in what I had, since vet school, and I'm not an immunologist, but um, I know more than the average undergrad, and I know what I think I know what they need to know. Most of them are, they're biology majors, so maybe they're thinking about professional school or nursing school or something like that. So sort of the clinically oriented angle of things. So I have no idea if I did a good job or not, but but I did. It was a nice sort of intro to teaching, um, and yeah, and it also made it that much easier for me to get the next position.
Mm-hmm.
Um, and so I lived in Pueblo, Colorado, with my husband, um, and um, he had a small research and development company that was ended up going out of business, and so I started looking for a full time job. And I'm trying to remember exactly how it happened. I got invited to, or my major professor was invited to do a workshop, and he said, "How about if I send Virginia instead?" And so I came to Texas A&M for a workshop, and it was related to feedlot and antibiotics, um, and I knew of a clinical pharmacologist, so I was boarded by then. I knew of a clinical pharmacologist who was at Texas A&M at the time, and I emailed her and I said, I'm coming to town. We've never met before. It'd be great if we could get together. I'm going to be here for a couple days. And my recollection is probably not completely accurate, but my recollection is she emailed back and said, "Oh, yeah, sure. That'd be great to meet. Are you by any chance looking for a job?" And so I came and kind of met a few people, and then I came back for a formal interview. And yeah, so that was I started in 2005. Um, as a clinical assistant professor, focusing on teaching pharmacology. But I also had a percentage. I don't remember what the percentage was at the time, but a typical faculty member has, we call it FTE. So, what's your percent that you are expected to spend on research, teaching, and service?
Mm-hmm.
And the bulk of my FTE was service. I mean, teaching, but I also, I had done a little bit of research, and I was interested in doing more. So when I got to A&M, the uh, investigator who had run the workshop that I came to said, "Well, come to our meetings. We're doing more work in this area." And so I sat in on our meetings. We collaborated on a couple things. Morgan Scott is his name, and then I met some clinical faculty, and I did a project here and a project there. I As the pharmacologist in the room, I could provide sort of that expertise on, well, if you'd like to look at drug disposition of a pharmacokinetics of a drug in a particular animal, I can look in the literature, see what we already know, help you decide when to take samples, what samples to take, and then I can do the analysis for you. So very much I was not in there, doing the animal work, collecting samples and that sort of thing, but I was providing that, expertise. Um, and so that's a lot of my research experiences on the teams of people. And I have this one little slice of the expertise, and we all come together. And I think you saw that, Tyler, in all the investigators that came during the research class last fall. So many people. That's um, Most research is not one single person doing something in a lab. It's people who bring in others with the different expertise to provide their, this is how we think we should do this. This is how we interpret this information, and so on. So that's been the majority of my time here at A&M is teaching in pharmacology. I teach a few things in a few other courses. Um, I had an interest from, when I was in vet school, certainly in grad school, in evidence-based veterinary medicine and the kind of processes associated with it, the approach, the understanding the literature, critical appraisal, all that kind of stuff. So I've participated in areas in various places in the veterinary curriculum, kind of not as a whole course, but, pieces of things. I have taught, actually, I just finished teaching an undergrad pharmacology course this semester that I haven't taught before. It was 2/3 undergrad, a third mostly non-thesis masters, but a handful of other graduate students. I've done a study abroad. I developed a course called Applied Pharmacology that I have only taught abroad. Um, so students were either in a semester long or a summer program, and it's a 2 credit elective, and it's very much the kind of what are the big ideas in pharmacology, what are some tools you could use that might help you in other fields, um, evidence-based medicine, reading the literature, understanding more broadly things like economics of drugs and drug regulations and political issues related to drugs. So kind of, I see it as clinical pharmacology, but I called it applied pharmacology because it's not a clinical level course. But um, and I used to teach a grad pharmacology course. It was sort of like a journal club-ish. Read the literature, write about things, and I continue to do collaborative research. We can talk about the dog aging project if you want in a little bit, but that's kind of my big um, chunk of my time now on research is related to the dog aging project. So I still do a few collaborations here and there. I have written quite a bit about um, teaching pharmacology. So in the university world, we sometimes call it the scholarship of teaching and learning, or writing about teaching. So I haven't done a lot of sort of formal teaching or learning experiments. There are people in our college that have done
Mm-hmm.
things. So Dr. Washburn and
Mm-hmm.
others in physiology, Dr. Patterson, have done some sort of formal experiments, maybe not randomized controlled trial, but sort of quasi-experimental interventions and measured sort of before and after, trying different teaching
Mm-hmm.
techniques or trying to understand how students learn or whatever. Um, and I have not done that sort of thing, but I have done a lot of sort of descriptive work on how we do this. A paper just got published last week actually, on how I collaborate with librarians to deliver content related to EBVM, and um, so so that sort of pedagogical interest in teaching, in addition to animal research, pharmacology research, that sort of thing.
I mean, all of that like kind of does make sense to tie together. Uh, Before taking the pharmacology course, I was, I don't know, when it was explained that we're gonna obviously learn about drugs, and then we had this EBBM thing that was somewhat tied to it, and I was like, why are these together? Now I kind of get it. You could like study, or you could do control trials or randomized control trials or any kind of study on drugs and like forever, because things change and there's always new drugs coming out, and drugs affect different species, and so in vet med you can kind of
Yeah.
explore the world with that. um, but you mentioned the dog aging project. What are you doing right now with that, or I guess first could you explain what it is?
Yeah, I am not the expert on the dog aging project. I'm a cog in the giant system that is the dog aging project. The dog aging project is the goal of the dog aging project is to study the environmental and genetic aspects of aging in dogs, um, because we care about aging in dogs, but also as a model for human aging. And it's a longitudinal study, not interventional, although we do have a clinical trial that is where we have an intervention. But for the most part, the project is designed to follow dogs throughout their lives and gather information on those dogs. Most of the dogs in the project, the information that we have on them will be survey based. So owners nominate their dogs, and then they complete very extensive surveys on all aspects of the dog, what they eat, where they live, where they sleep, things like, are there stairs in their house, how often do they get walked, very extensive surveys about their environment, um, and then we ask that survey every year. So what's changed, and then describe, behaviors of your dog. If you have medical records that you can upload, can you upload your medical records? That sort of in infancy stage is evaluating the medical records. We have some individuals that are working on some artificial intelligence approaches, machine learning, really approaches to like extracting data from records.
Yeah, I was wondering, I was like, could you use AI at
Absolutely.
all for any
Yeah,
of that?
absolutely. Yeah, and unlike in human medicine, there is much less consistency across medical records in
Yeah.
for animals. If you're in a big corporate practice, then there'll be consistency across the practices, but across all the practices in the United States or around the world, but for sure in the US, there are still some practices that have handwritten records or a homegrown electronic medical system. Um, and often the electronic health, whatever the electronic medical record is, its main job is as an invoicing and an inventory tool, not as a medical record of the dog's or the animal's life, health, whatever. So yes, that is an ongoing wish. And what can we and how accurate is it? And then could we then ingest lots of records and really get in depth in things like things I'm interested in pharmacologically, right? Like how well do dogs respond to various things? Can we use sort of like a huge cohort study, right, of a prospective or retrospective analysis of dogs responding to various drugs, right, where you have medical records of when they started a drug and what their health was like after that, or vaccines and all those kinds of things. So, but a lot of that is happening in human medicine, and I think we're poised to do a lot of good work in veterinary medicine as well. Um, so back to the project, the majority of the over 50,000 dogs are, we have survey data on them, owner reported data. And then there's cohorts within that where we collect additional samples, for example, blood samples, and where we look at DNA and evaluate various genetic factors. And so we're really looking at um, marrying up the genetic data with how long do dogs live? Is there a difference based on where they live, what size they are, um, what breeds they are, all those kinds of things? Can we correlate those things, or can we associate various things with aging? And are there biomarkers that we can look at longitudinally to say, if when a dog was 3, if it's some protein was elevated, then that predicted that they got cognitive dysfunction at 10, whereas if it was lower, it didn't, where that's the goal, is to be able to describe but also predict, and understand and potentially translate to humans what factors are associated with good aging or bad aging, meaning longevity generally speaking, but also what's called health span. So not only do you live longer, but does your dog live longer? But are they also healthy for longer?
Hmm.
Just living longer isn't necessarily a goal, right? You want to
Yeah.
also be healthy while you're living longer. Um, so yeah, it's I don't I can't remember the number of institutions, more than 10, um, probably more than that if you include all our clinical trial sites. Uh, and interdisciplinary. So pure geneticists, immunologists, people who study dog cognition, um, veterinary neurologists, veterinary cardiologists, um, people with expertise in aging and Alzheimer's disease. Their cognitive dysfunction in dogs is a pretty decent model for Alzheimer's disease in people. Um, and probably things that we don't even know yet, right? That we're in part of the descriptive access approach and longitudinal approaches to see if there are things that we don't know about that we can also find, and then investigate more experimentally or in laboratory animals, or and then translate to people. So the cohorts include this sort of small groups of dogs that are pulled out for analyzing more um, biospecimens, so blood, but also urine and feces, so microbiome, and how does the microbiome change over time? How is that associated with aging and other things that happen to dogs? And then we do have a clinical trial, the Triad trial, which is the trial of rapamycin in aging dogs. So rapamycin, sirolimus is another name for it, has been shown to extend lifespan in a number of laboratory animal species, flies, rodents. Um, and so, is that true in dogs? We have some small, There are small studies that have shown that it can make a difference, and that the dose that we use is relatively safe compared to the dose that's used for other conditions. So the drug is an immunosuppressive drug. It's used in organ transplant patients at much higher doses, but a very low, very infrequent dose. Um, so once a week or something like that, where you don't have where you're not concerned about the immunosuppressive effects, because the central tenet of pharmacology, right, is dose matters, concentration matters, and so regimen is really important. It's not just that the drug is there; it's how long is it there and at what concentrations. So, and we are in the process of recruiting dogs. Some dogs have already completed their whole study period. We treat them for a year. They are treated for a year by their owners. They fill out monthly surveys. They have every 6 months they get a visit to one of the clinical trial sites, and a bunch of samples are taken, and we evaluate a lot of different things. There's a group of animals that we're evaluating cardiac function, and a group of animals that we're evaluating neurologic function. Um, there's a new drug on the that's been conditionally approved for cats. It's the same. It's rapamycin for cats with heart disease, and so we know that cardiac function is affected by rapamycin. Um, and so we follow them for 2 years after the end of their 1 year treatment period. So it's 3 years on the study. So it's going to take a while to, um, but the NIH funded the project because of its translational impact on
Hmm.
human aging, and the other things that we're going to be finding out along the way about how safe the drug is, how well it works, what it does.
I was going to ask what the difference was, or the distinction between a clinical pharmacologist, and you said an experimental
Yeah,
pharmacologist.
yeah, or what some people just call a pharmacologist.
Hmm.
So, um, m- my personal sort of definition of a clinical pharmacologist the is someone who integrates all of the factors related to a drug or group of drugs or drug therapy. So it's understanding how drugs work. So at the molecular or cellular level, what is the mechanism of action? What you memorize in vet school, right? Like, what are all the mechanisms? But then what does that look like? So just because this thing happens in a cell, what does that mean for the organ that cell is in, or the tissue that cell is in? And then what happens in the individual animal person when you administer that drug, um, physiologically, and also all the other things associated with decision making about drugs. How are drugs different from each other? What's the evidence to support one drug versus another in a particular setting? That's where the EBVM stuff really sort of dovetails with pharmacology. So it's not just is this the mechanism that we think might be useful, but if we give this drug to animals, does the thing that we want to happen actually happen? Or in what percentage of animals does it happen, right? Because not every drug works every time in every individual. And what dose does that thing happen? And then can we predict which dose makes that thing happen? And that's pharmacokinetics, which is a kind of one of the if you list all of the things that most clinical pharmacologists have some expertise in, they might not be a pharmacokineticist, but they understand pharmacokinetics, and they can most of them have, at least at some point in their careers, learned how to understand pharmacokinetic modeling and study design and things like that. Some clinical pharmacologists, that's all they do. Some it's what they did when they were in grad school and they don't do it anymore. So to me, clinical trials is the purview of a clinical pharmacologist. Um, Conduct, study design, compliance. So when I was in grad school, a pharmaceutical company sponsored my project, and it was pre-approval, so it was before the drug was on the market, and so the company very was involved in all of the study, and they had study monitors, and we ran the project as a GLP GCP, so good laboratory practices and good clinical practices, which are regulatory requirements for performing studies for drug approval for humans and animals. So it's about um, having standard operating procedures, always doing the same thing, keeping track of who did which things, calibrating things, so that when you say, well, their temperature was 102, how do you know it was 102? How do you know it was incorrect? So it's very close um, monitoring, keeping track of things so that you could recreate the entire study if you needed to. You could look at every aspect of it. And so that's another thing so the sort of regulatory aspect of how do drugs get on the market, drug development, how do you discover drugs, and then how do you develop them into actual products, clinical trials, pre or post drug approval, and then moving across species. So the comparative aspect, most clinical pharmacologists that I know have at some point in their career, maybe not in their late careers, but earlier in their careers, knew a lot of comparative pharmacology. So lots of veterinarians, maybe they don't specialize as in go to a specialty, take specialty boards, but they focus on, one or two species. And so um, they get really comfortable with the drugs that are used in dogs, for example. But the rest is, whereas the clinical pharmacologist, especially one like me who teaches at a veterinary school, like maybe I'm not an expert in all of them, but I have enough information that I can um, talk knowledgeably about, the majority of drugs that are used in most veterinary species, as opposed to someone who's a small animal internist or a large animal surgeon, where they're going to focus on, the drugs that they use in their area. Um, so to me, a clinical pharmacologist is pulling all of that information together. It's also, and I don't do this very often, but I know I have colleagues that have the same specialty boards who do this regularly, which is uh, at the patient side, making decisions about um, like therapeutic drug monitoring. Do we need to change doses on this drug because um, the the patient is in kidney failure or something like that? So it's um, applying drug decision making actually at the individual patient side. I do occasional case consults, especially about antibiotics, and particularly in resistant infections. Well,
Mostly in large animal?
no, both sides.
Okay.
So both large and small. I work very closely with Dr. Sarah Lahan, who's the clinical microbiologist, and we talk frequently about cases um, and somebody has a complicated infection, or they've tried some drugs, or they got a panel back from the lab and everything's resistant. Now what do we do? Or is there another drug we can test? Or things like that. So it's managing patients, um, managing therapy kind of at the patient side, and that there are clinical pharmacologists that do that as well. So
You mentioned that you did a project for a company um, testing out their drug, I guess. Pre like I guess early on in that process, what was that like? Because there's kind of a lot of opinions about the drug approval process, or
sure,
like
yeah,
like I guess drug company world.
sure. Yeah, and I just got a glimpse of it, right?
Hmm.
So, I mean, I have pharmacology colleagues who work in industry, and I don't, and I've only just have a glimpse of their world and how that works. And when I did this project, I got a glimpse of that world, and it's a very rigorous, structured process, and the requirements um, that are transparent, publicly available with the FDA, by the FDA, Center for Veterinary Medicine, or if you're in Europe, the European Medicines Agency, or it could be the individual, like Health Canada, Veterinary Drug Directed, or whatever. Um, they are very transparent in most cases about what it is you need to do to demonstrate. There's two things: safety and efficacy. Safety meaning not that every drug is uh, never causes a problem, because that's not the case, right? All drugs have some potential for causing an effect that you don't want. Um, but can we predict, if we give the drug to enough animals, can we predict what percentage of them may have, an adverse effect? What are those adverse effects? What do we need to monitor for in patients that get the particular drug? Or how risky is the drug? The riskier the drug, the more we need the payoff to be high, right? So chemotherapeutics are the classic example of that. So cancer drugs, um, most cancer is really bad. We are willing to risk some adverse effects because we want to treat these diseases that are, potentially fatal. So, whereas if we're treating a skin infection that's relatively mild, we're not willing to put up with a lot of adverse effects in that particular. So, safety: how safe is the drug? Is it, and is it safe in that particular species of animal? So, lots of, there is often work, some of it's in vitro, tissue culture and things like that, looking at drug safety and how does the drug work. But you actually have to put it in an animal, a laboratory animal, or um, some other kind of animal, to see what happens at the whole system level. And they're very strict, regimented requirements that are very transparent. And these days, most companies, have meetings with the FDA personnel to say, okay, here's what we want to do. Does that sound reasonable? Are we going to get the answers that you're interested in? Are we going to be able to, address the questions that are of interest? So, safety and efficacy. Efficacy means, um, that whatever it is you want to put on the label, you've demonstrated that that's what's going to happen, at least in more animals than that don't get the drug. So the comparison between animals that don't get a drug and animals that do get a drug, there needs to be a difference to demonstrate efficacy. That difference doesn't need to be 100%, right? So it doesn't necessarily mean that the drug works in 100% of animals, and if all the animals that get it, um, have whatever the desirable effect is, and zero animals have the desirable effect, and if they don't get the drug. But there needs to be a difference, and a statistically significant difference. Um, and so there's that safety and efficacy. And then what I was talking about earlier, which is how the conduct of how all of these studies need to go, and that's, um, some of that is outlined in the GLP GCP process. So, good laboratory practice and good clinical practice is where we can recreate exactly what we did in the lab. Or in the GLP would be, things like, diagnostic assays. Like if you're doing CBCs or something like that, you would do that in a lab. So that'd be GLP. GCP would be, how are you handling the animals? Um, have you gotten, owner permission, informed consent, and those kinds of things. So those are all very, explicitly laid out in a number of documents that the FDA publishes. Um, the They call them, guidance documents, and so, uh, it's a rigorous process with a lot of oversight, and a lot of back and forth between. It's an unusual these days, at least from my understanding. And again, I don't work in industry, so this is my understanding, is that it would be unusual for a company to spend 10 years on something, never have a discussion with the FDA, show up with a bunch of data, and then have the FDA say nope, because they will have had conversations along the way. That's not to say the FDA doesn't say no. They might say, well, you expected this product to do this, and it actually didn't do exactly what you thought it was going to do. And they might say no. Or they might say, well, that's actually not enough difference between, the treated group and the untreated group. Um, and um, so I think it's as good a process as it can be. The thing I think that is, can be hard to understand, and it's definitely hard to predict, is if I do, it's a relatively large studies that are performed for drug approval. So 4, 5, 600, 1,000 animals that get a drug. But now once it goes on the market, now it's going into thousands or tens of thousands or hundreds of thousands of animals. And so it can be hard to predict, like, are there some adverse effects that are maybe not didn't show up in that first, because it was a, more or less a random sample of 1,000 animals, and now I give it to 100,000 animals. They're bound to be animals that look different than the 1,000 animals, that have other conditions at the same time, that and potentially, if it's a rare adverse effect, if you only do 1,000 animals, it's not gonna show up
Hmm.
necessarily, right? Or it might have been one animal. Whereas if you put it in a million animals, now you've got, a number where there's gonna be a signal. So um, I don't know if that answered your question or if that was kind of where you were going.
Yeah.
What do you think the sort of what do people ask about, or what do people are concerned about
I mean, I
in
don't
drug
know.
approval?
There's a lot of opinions about the economics with medication and stuff, but I was mostly curious with the FDA's, since I've been in college in undergrad as an animal science major, the FDA and the USDA have been looked at very fondly. It's like these are very robust systems that we have
Yes.
for drugs approvals and for food production.
Mhm.
Um, I don't know if you know the answer to this, but you mentioned Europe and Canada. I guess anything outside of the U.S., is it similar there?
Yeah, that's my understanding. There are some agreements that where a group of countries, so like Canada, the U.S., Australia, Japan, and can't think. I'm not sure which year. Maybe it's the European Medicines Group, where they will all come together and kind of hammer out an agreement that says, if you we're all going to agree that these are the things you need to do for this particular aspect of drug approval, so that it makes it easier for a company to say, well, we did it in, not that it's a, if we got it approved in the US, so you should go ahead and approve it in Japan or whatever, but that we did the studies that you wanted, so we don't need to redo the studies because we were all doing the same studies,
Mm-hmm.
or they all look the same.
Because then you waste money doing the same
Yeah, and
thing.
that still happens. There's not agreement on everything, and it's not agreement around the world. But it's better than it used to be, I think.
Does it ever happen that it one group in one location does one study and then another group does something maybe not exactly the same but similar enough to where when the results come out and they're different, then people start questioning the results themselves?
So like for the same drug in both.
Yeah,
Yeah.
yeah.
I mean, that happens. It probably wouldn't be for drug approval, because if you are if you're putting a drug on the market, then you have a patent exclusivity for some period of time, right? So
Mhm.
if you have a specific drug in a specific formulation, then you have some protection, at least in the U.S., for that, drug and formulation for some period of time, depending on when got all your when you first filed the patent. You know how long that is. Exclusivity is what it's called, and so it would be unlikely that another company would have the exact same product,
Mm.
brand that would be another branded product. Generics are a different story. We can talk about that separately, um, um, but a product might come on the market, and then somebody like me in an academic institution or at a teaching hospital might say, "Well, let's see what happens in our patient population," and the effect of the drug might look different. Maybe the treatment effect is different. Maybe a fewer percentage of animals respond. But that's kind of the nature of experiments, right? You do an experiment in 1,000 animals, and then you do the experiment again in a different 1,000 animals. You're not going to get exactly the same answer or the exact same percentage of animals responding. And if you don't get the exact or a similar percentage, you start thinking, well, is it us or is it them, and why? And this is actually where lots of um, additional investigation then starts to happen because someone says, well, wonder why that is. Is it a breed thing? Is it a, this is how we figured out um, things like MDR1 and, peak glycoprotein because there was a subset of animals that had this weird thing happen. So what can we tell about them that makes them different? Is it a genetic? Is it a, organ function thing? What is it? Um, and so, but you would never expect that if you do the same experiment 3 times on 3 different populations of animals, that it would look exactly the same. But if it's radically different, you start thinking there's something. My inclination is not to go, well, it must be the pioneer drug. They didn't do a very good job with their studies. No, it's something about the environment was controlled in a different way. It was dogs in Massachusetts and now we have dogs in Texas, or it was golden retrievers and this study was all in Irish wolfhounds, so maybe there's something going on there that modifies the effect of the drug enough that it looks different in a different subset of animals. So I don't is that kind of what you're getting at?
Yeah, yeah. Because I can imagine that if you run multiple studies on the same drug and I guess, recreate a study, and then you get results that are different enough to cause, I guess, questions about maybe the results weren't measured correctly, or
Sure.
I don't know. I don't know. You would have to speculate on what to do next. Um, but I guess it sounds like you kind of you do enough with the drug that um what is it? What am I trying to say?
Well, I mean, I think you if you so in pharmacology, we talked about estimating treatment effect, right? Where you're comparing the result of if I give the drug, if I give a placebo and I give the drug and the percent response in each of those, and the difference between the two is what difference the drug made. And we call it an estimate of the treatment effect because you're basing it on one single study or maybe 2 studies that you looked at, because you can't look at the drug in the entire population of animals because that's not possible. And also, you can't afford to look at it in 100,000 dogs. You can only afford to look at it in 200 dogs. And so we call it an estimate of the treatment effect. It's not the actual treatment effect, it's the estimate of the treatment effect because it's a sample of the population and we're making an estimate. If we did another sample of the population, we'd have another estimate. And the more samples we take or the bigger the samples, the more precise our estimate is. But it's still just an estimate because it's like any other experiment, right? You're only taking a small portion of an entire population of things, whatever it is, animals, cells, whatever. And so it's always only going to be an estimate of whatever that measurement is.
Mm-hmm.
Yeah. So I mean that's like the scientific method, right? Is and how science works is um we make estimates and then we find out other things and we revise that estimate a little bit based on, or we say, well, in dogs that are in sight hounds, it actually looks like the response looks different, and this is by how much because we did 10 studies or somebody else did. That's the essence of meta-analysis, right? Is pulling together data from multiple studies
Mm-hmm.
and in a statistically valid way, pulling those together and saying a more precise treatment estimate because now because I have 5 studies in 100 animals instead of 1 study in 100 animals. Now it's like I did a study in 500 animals. That's what a meta-analysis is giving me. So now I have it's a bigger picture of the population. It's more animals, but it's probably also more diverse animals. So different breeds, different ages, maybe different severity of disease. This is often what happens in so the drug the studies that are done for drug approval, part of the control of the experiment or the study is the clinical trial is the animals can't have other diseases. Well, when you bring that out into the population, now we have animals that have other diseases. So are the other diseases the issue? Are they modifying the effect of the drug? If a dog has atopic dermatitis and dermatophytes, right? That's different than if the dog just has atopic dermatitis, it's likely to respond differently to the drugs. So that's the real value of there's the really controlled environment of the drug approval studies. And then there's the how do we understand it in animal populations? So the value of clinical studies in once a drug is on the market in multiple parts of the world in multiple settings in, you know, primary care versus tertiary care, all of those things are really valuable to help us get a full picture of how well does a drug work, what is its true treatment effect, and what modifies that treatment effect.
It sounds like the system kind of protects drugs that once, I guess, at some point, a chemist or a experimental pharmacologist would create a drug or develop it. And then throughout the process, if something is flagged, maybe an adverse effect or um, something isn't as ideal as you would imagine, uh, it isn't just cut off right there. It's like, okay, let's figure out what's going on. And then the multiple studies kind of help with that because what is it, ivermectin and MDR1 you mentioned earlier? It's like if at some point found like we didn't know the MDR1 genetics existed
That's
and
right.
we just saw the effects of ivermectin
That's right.
at those concentrations, it would be disadvantageous to just say ivermectin is not good for dogs because now we can use it either at lower concentrations or knowing it for these dogs to watch out
That's
for
right.
specific things.
That's right. It's precision medicine is what it is.
Hmm.
It is the beginning of the promise of precision medicine is if we can understand how subpopulations, subsets of animals have different whatever it is, and most of it comes down to genetics or epigenetics, whatever the difference is, whatever receptors or whatever, um, and identifying if we can say if they have this subset of things, they are less likely to respond, or they are more likely to respond, or they are more likely to have adverse effects. We know in human medicine, they actually have a lot of more information about that sort of thing. So there are drugs that we know are interfere with or inhibit or are metabolized by the CYP proteins. So phase one protein enzymes in the liver that metabolize drugs. Um, and there are drugs that we know are affected by that. So individual people who have a particular allele at that, the gene that encodes for that enzyme, where that enzyme doesn't work as well in some subpopulations of individuals, they are slow metabolizers of the drug. So we either don't use that drug or we give them a lower dose. Or there are fast metabolizers so that we either don't use that drug or we give a higher dose. So there are a number of drugs in humans where we actually know those things, and we can modify, either choose different drugs or change how we dose the drugs. We just don't have that kind of information or the resources to figure out what it is about why some dogs don't respond as well as other dogs do to specific. I mean, I think we're doing more of that now than we ever have, but it's a compared to what goes on in the human world. It's We're still sort of in the infancy of sort of figuring that out to be able to say and to have the diagnostic test, right? To say we're going to do this. We're going to take a sample of your blood. Or your dog's blood, and it looks like they have this gene um, type, which means they're going to process this drug more slowly or more quickly, and therefore we should do something different. I mean, we're doing that definitely with the MDR or the ABCB gene, um, where there are a number of drugs. So Washington State has a really nice website where you can go and see like all the drugs that we know are substrates for the ABCB1 um, um, transmembrane protein. And therefore, if you have a dog that has a mutation in that transporter protein, you should be careful giving them these drugs because those drugs are going to cross the blood-brain barrier, or they're not going to get absorbed well because those ABCB genes are also those transporters are in other places too. They're in the GI tract and other places, and so but we got to have the clinical evidence. So there's the experimental, like we can clone the gene and figure out where it is and what it does. But then we might not know until we actually give the drug to a group of animals what the outcome of that mutation is. So we worked backwards. Um, Dr. Katrina Maley worked backwards from we have a subset of dogs that responded very badly. Many of them, some of them died from ivermectin toxicity. What is going on? First, we thought it was a phenotype, right? White feet don't treat. That was
Mm-hmm.
the mantra because it happened in Australian Shepherds and Collies, and but then the genetic analysis suggested that um, led to the discovery of this mutation in the P-glycoprotein, which meant those dogs, the P-glycoprotein, that transporter, ABCB1 transporter in the brain, is really important for protecting the brain. It pumps stuff out. So if that pump doesn't work, then drugs can get it. Certain kinds of drugs can get in there. Um, Chemotherapy drugs, we have there's some opioids that also use that same transporter, and so we have some pretty good information about how to modify, either don't use those drugs or modify their dosing in those drugs, or use different combinations of drugs um, to help prevent those adverse effects in those subsets of individuals. So the promise of um, and some of this is something that is another sort of potential of the dog aging project is we have we have biospecimens, including the basically the whole genome of 1,000 dogs, and then we have health records that we can tie to what's happening to the dogs, what drugs have they gotten, whatever. I mean, there's a lot of potential for kind of figuring out what modifies drug action in individual dogs. So.
Yeah, I was thinking a little bit earlier that farther down the line, maybe at some point, because there are those tests that you can send your dog's DNA, and it's like, oh, what breeds are they? And
Right.
at I'm kind of like skeptical sometimes. Like you get a mutt, and they're like, well, it's like 30% this and
Yeah.
30%. And I'm like, what does that really like? Does that help at all for whatever? I don't know. Maybe it's kind of cool. People enjoy it, but um, no. At some point, you could use a genetic test and just screen for a number of things, which in human medicine they do, which I don't think animal medicine's ever going to compare to human medicine. Like it's just what we do for animals is it's it matters to the extent that the animals matter to us, and animals matter to us a lot, which is why we do so many different things. But at the end of the day, I think people will choose people over animals if that was ever a choice. Not saying it is a choice now, but that's certainly how we treat medicine in today's world. Um, and I don't know if that'll ever change. But when it comes to adverse effects, it seems like they're mostly stumbled across instead of predicted beforehand. Because I can't imagine if you knew that something was going to have an adverse effect that you would go ahead and like not try and prevent it ahead of time. And drugs are complicated enough; you're not really going to know what all it's going to do before actually running those experiments. But when uh, when coming across a adverse effect, um, what I guess what is that like? Like how is it first presented? Is it like the oh, we keep seeing these dogs come in and it seems like does someone down the line just say, "Oh, I see a pattern here." Like is it just that kind of luck?
Yeah, that's a great question. I think, let me first say that, there are different types of adverse effects. So there are the adverse effects that you would absolutely predict because of the pharmacological effect of the drug, right? So if a drug is a um, beta agonist, then you're going to expect that, and you're using it I don't know, elevate heart rate. You're also going to expect that will have other effects that are related to beta agonism, right? So there are pharmacological adverse effects that are predictable. It's the idiosyncratic. It's the things that it's like in people, the antibiotic fluoroquinolones, or the group of antibiotics, fluoroquinolones, have been associated in people with tendon rupture. Well, that is not at all, at least on its surface, predicted from how the drug works. Not certainly not how the drug works in a bacterial cell, right? That doesn't, like, what does that have to do with tendons? Um, Mechanistically, it could actually be explained, probably. Um, and sometimes that happens where we work backward to say, well, this is a potential adverse effect. Why is that happening? We don't always do that. Sometimes we just say, well, it's an adverse effect. We need to worry about it. But so how do those signals come up? So there are a couple of different ways. One is we were talking about this earlier, I think, uh, a drug company that has a product on the market is required to report to the FDA any information they get from users. And the FDA also um, passively collects that information. So if you treated a dog, you're out in practice and you treat a dog with a new drug and something happens, the dog has a seizure and it's never had a seizure before. Your part of your role as a veterinarian is an obligation to say, that's weird. This is not a drug that should have caused a seizure. That's not on the label. I don't know what's going on. Maybe it's the dog. I don't know. But I'm going to report it to either to the FDA or to the company. And then if the company got 1,000 reports in the first month that the drug is approved of seizures in dogs, they'd be like, huh, what's going on here? That's a signal. They would investigate that. There's a couple of pretty good. It's been a while now, but the FDA wrote a paper, or some individuals at the FDA wrote a paper, some years ago now, that kind of described their approach to. We got reports about these things. We got, you know, enough reports in a short enough period of time that it seemed unusual, and therefore we investigated. We evaluated what other drugs the dogs were on, or the animals were on. I can't remember what species. We evaluated, the medical records of the animals. We questioned the veterinarian. We, et cetera, et cetera. And um, all other things being equal, we think this is potentially related to the drug and not to something else that was going on in the animal. Because that's the challenge, right? Always is. I mean, um, the most believable, accurate, I suppose, way of determining whether something is related to the drug or not is you give the drug, you get the effect, you take the drug away, the effect goes away, and then if you give the drug again, you get the effect again, right? That's kind of the
Consistency.
yeah, and but that just doesn't that's not always possible.
Hmm.
Maybe you don't want to do that. Maybe you don't want to cause more seizures in a dog. You don't want to take the risk of that. And so we don't always know. But you sort of rule lots of other things out. And then what's the percentage of animals that are responding, and are there differences across the animals, or do they all look the same, and that sort of thing? So that's um, it's it, it's sort of passive surveillance, so reports of things that are unusual. And I mean, that's kind of how scientific investigation works, right? Something unusual happens and you go, huh, I wonder why that's happening. Let's look further. Let's
Hmm.
hypothesize. Let's test that hypothesis. And so that is my impression, at least, of how we sort of discover or enumerate what additional adverse effects are than ones that are, found in initial investigations of a new drug, or a drug that is being used for a new thing, right? In veterinary medicine, um, we often use drugs that are approved in other species like humans, and now we use a human drug in a dog or a cat, or dogs and cats, and we maybe they have different adverse effects than what happens in humans. And so, yeah, we discovered new things that way, and then try to figure out whether it's the drug or the something about the animal or the disease condition that caused that, or um, was mediated by the drug versus the condition, or the animal or the species, or the breed, or all the things that has impact on physiology.
Yeah, that science, like the scientific process, is really fun when you think of it
Yeah.
like that. Like, oh, what is
Says the
going
budding
on?
researcher, right?
Yeah, yeah. Well, over the past 2 years being in vet school, I've come to enjoy the science part of I guess, my education a lot more. When I was a lot younger, uh, I knew a number of people who were like, "Oh, science is amazing. It's so much fun. You get to discover all these things." At least when I was in that position, as a kid, I was like, well, I'm not going to do anything right now with it. Like, it's a thing. And if anything, we were learning very basic things as I was growing up, things that really haven't changed in a long time. And I was like, it's so far away. Like, it's really not that interesting now, especially going into this summer research thing that I'm doing. I'm like, oh, this is like, I can actually do something now. I can be a part of something, and maybe something will come out of it. And I don't know, it's fun. And kind of going back to your whole route of getting here, like, did you always know that you were gonna be a teacher?
No. I mean, there were some maybe inclinations toward that, or things that people would say to me, not so much, oh, you should be a teacher, but sort of my persona in various jobs that I've had. My my dad was a college professor, so I didn't know what he did, but I
Hmm.
saw that sort of academic life.
You're exposed to it.
Yeah, yeah, and most of my friends' parents were college professors too, so I grew up in a small town that was a college town.
That's kind of fun.
Yeah,
Like,
so
your parents and all of your friends' parents are all teachers.
yeah, and so um, and my mom did some kind of volunteer teaching, and so it was sort of in my um world. Um, but I never, when I was in college, I went to a small liberal arts college, Kalamazoo College, and I was a psychology major, and I didn't really know what I wanted to do, and I never thought, oh, I really want to be an academic, or I really want to be a teacher, I want to be instructor. That really wasn't. But I did like the academic. I mean, I did like the academic environment, and I mean, fast forward to end of vet school, what am I going to do? I was really interested in in food animal, and but I didn't have a lot of experience in food animal. I'd worked on a dairy farm a little bit, and I spent a couple of lambing seasons at Heifer International
Hmm.
in Arkansas. It was Heifer Project at the time, but they had a kind of a demo ranch and they had a bunch of sheep. And I spent a couple of volunteer spring lambing seasons there. Got really, I thought small ruminants are cool. Animal agriculture is cool. Um, so after vet school, I decided that an internship, a food animal internship, would be a good segue to getting a job in private practice in a food animal with predominantly food animal. I'd have more experience if I did an internship. Um, and in my internship, I did a lot of, I did clinical teaching, right? I had students in the truck when I was in the truck, or we did the sale barn every Friday. So we had students would go out to the sale barn with us, and um, or in the hospital, and I liked that a lot. Um, and then I was a clinical instructor for a year, and it was the same, very similar. Mostly it was field service, ambulatory stuff, and I enjoyed that. I didn't do much of didactic teaching, but it was, watching students figure out how to do things, learn things, go, oh yeah, now I understand why that goes together with that. Now I see the application of that. Um, or oh, I never thought that would be interesting, and now I do. That kind of thing it's fun.
Hmm.
Um, and in in a vet school, especially in those days, um, being a veterinarian without any advanced training, you were not going to get a job in academia. That's changed a little bit, but it's still most positions require specialty training, like
Hmm.
specialty boards, or a research degree of some kind, master's or PhD or something like that, depending on what the job is.
I mean, I can't imagine anybody would get hired at the university now. With like, specialties are, they're kind of getting more popular as time goes on.
Sure, there's a lot
Um,
more specialization. Yeah.
I mean, it, at least in small animal medicine, that's kind of the world I live in or grew up in, it people are more willing nowadays to do more for their animals, and so the feasibility of people of these specialists being out there in private practice, and with corporations growing, it I don't know, it just makes a lot more sense nowadays. Like, uh, yeah, in a university setting where we are teaching the new generation of veterinarians or people in medicine, you would want those people who are like, this is what I do, this one class, like the pharmacologist teaching pharmacology. You wouldn't want just like a private practice veterinarian being like, all right, now you're going to teach immunology to these veterinarians who are about to graduate, and then do this on their own.
Right.
Um,
Yeah, and I mean, I think depending on what content you're talking about, there's different levels of expertise that are necessary for a an academic institution to say, yes, we believe that what you will do, what you will teach, how you will do it will is adequate and is will get us to, we need to graduate graduate veterinarians who are capable of, day one practice. Um, yeah, and definitely in when I graduated from vet school, the specialists were in academic institutions. There were a couple of big specialty hospitals like in the country, but you know, there wasn't the specialty hospitals, private practice specialists that exist now across the country. Um, or the, cardiologist who goes from practice to practice,
hmm
or the radiologist who reads films from home, right? Those didn't really exist when I graduated. And there was the um, traditional vet school. The pharmacologist teaches pharmacology, the immunologist teaches immunology, the internist teaches medicine, the surgeon teaches surgery. And we didn't have most schools didn't have the kind of professional and clinical skills courses
hmm
that you guys have and that most schools have now. We didn't have communications training as a separate entity. We didn't have critical thinking as a separate entity. It was, uh, physiology, microbiology, pathology, pharmacology, medicine, surgery, clinics, right?
hmm
Um, so the general feeling was if you were going to teach, pharmacology, you were a pharmacologist. If you were going to teach medicine, you were going to be an internist with specialty boards, et cetera. And and a lot of those specialty boards, especially in those days, did have at least a master's degree, sort of requirement. So at least some research exposure. And that's not there are a lot of um, residencies now where they don't, they might do a project, but it's not a full on graduate degree.
hmm
Well, a project that you do, your second, third year, whatever of your residency.
Why would
Um,
some require a project and some not?
why?
Yeah, some like the different between the different specialties.
That's a great question. I don't know. I mean, the every board has its own, is its own entity.
hmm
And so they determine what do we think a surgeon should look like? What should all surgeons be able to do? Here's what we think they should do. So therefore residency should look like this. And this is what we're going to test them on and, whatever. So every specialty is going to, think about it a little bit differently. Um, and that, I mean, it's sort of an interesting philosophical question. You know, do you think that a small animal internist or a small animal cardiologist is better who has completed a research project and a master's degree or who hasn't? Like, how do you identify what the skills are that they learned that might or might not be necessary to be an adequate cardiologist?
hmm
I don't I'm not suggesting that they're doing it wrong. I'm just saying like there are different ways of thinking about
hmm
what is important. What are your if you're running a residency, you think, well, what are the graduates from this program? What do we think they're going to do? And how do we prepare them best for that? If they are all going to go into private specialty practice, what are the skills they really need to be good at that and to be, specialists. Um, and I think thinking has changed over the years. My sense was, when I graduated from vet school and was thinking about specialty training, um, everybody did research, some kind of research degree at the same time, or some kind of research project that became a master's degree, maybe more. And I think part of that, I think, is because they were all at academic institutions. I mean, there are a lot of residencies now that aren't even at academic institutions, right? So they're at academic institutions where the focus is on generating new knowledge. That's what research is, right?
Mm-hmm.
Generating new knowledge. And so it's sort of automatic. Well, of course, if you're going to do your residency training in an academic institution that is, designed to develop new knowledge, of course, you're going to learn how to develop new knowledge along
Mm-hmm.
with learning how to be a cardiologist, right? Or whatever. And I think the recognition at some point along the way that maybe it maybe if you spend all that time doing a project that then you never do research again, is that really the best use of your time in a residency when you could be focusing on, clinical skills and knowledge and so on?
Mm-hmm.
I mean, everybody has a different idea about whether that's the case or not. But I think it's an interesting question to say, and it's an important question in any educational setting is what are we trying to create and are we doing that? And if we're not creating what we want, what should we change? And if we, how do we know we're creating what we want? Right? They pass the exam or they write the papers or whatever. And so I think it's a question. I mean, we ask that in the vet school, and every vet school asks that all the time, especially every 7 years when we have to be accredited. What is it that we're trying to create? How do we show that that's what we created?
Mm-hmm.
And are we doing a good job at it? And so if you're what you think you need to create changes over time, then then of course your what happens in that educational setting, whatever it is, also changes over time. So residencies have changed over time because they recognize that most of the um, individuals that graduate from the or you know finish those programs weren't going into academic settings and didn't really need that research credential. And maybe they felt like it didn't really actually enhance their training. I think there are always ways. I believe every vet student should know something about research,
Mm-hmm.
um, because even if you're never going to do research yourself, you understand where things come from. You understand what good evidence is and what bad evidence is supporting the decisions that you're making as a clinician. Not everyone agrees with me, but I think you can also spend a lot of time learning about quote unquote learning about research, but actually not learn anything useful.
Mm-hmm.
What's the point of that? That's a waste of effort. Um, so yeah, I think carefully thinking about why you're doing the things that you're doing and why you're including things in training programs and whatever is really important. So, but when I was finishing vet school and was in my internship, and I kind of realized that I don't know that I mean, I definitely liked teaching the kind of teaching that I was doing. Clinical teaching is super fun. It's most of the time it's very immediate. When I stand up in pharmacology and I talk and you talk and we learn some things and then I give you an exam, most of the time there's not like a sort of aha. Sometimes it happens, right? And
Mm-hmm.
it's great when it does, but most of the time you're like, yeah, yeah, yeah. Okay, give me the. Yeah, I know some more stuff now than I did before.
It would be nice to have an aha moment
Right?
8 times a day, every day for
Yeah,
an
yeah.
entire semester. But
That's right. And so I mean that for sure it happens, right? Particularly if it's a challenging concept, right?
mm-hmm.
And somebody comes up with a really good analogy, or someone asks a question that um, where the answer illuminates better than the pre than and you can see like I love to see that out in the audience where the audience in the classroom, right? Where someone will go, like do a oh, that
Mm-hmm.
like that's cool, but it's still you know it's in a sort of sterile setting in a lot of ways, but you're in a surgical suite, you're out on a farm and you go, you know what, if you hold it this way instead of that way, or if you cut here instead of here, or let me show you how to,
Mm-hmm.
and then like it changes. instantly what they're doing in the moment, and or you see that sort of like, oh, that's why we learned how to do it that way, because this is what, how it what what how it makes that work. That's super cool. So, but clinical teaching, um so that was interesting to me. But I also didn't really, there wasn't a clinical specialty that I really was excited about. I don't really like horses. I don't like being around horses very much. I think
You
they're really interesting, but I don't like, I wasn't around horses growing up. I feel like
mhm.
um there's some things in life that if you're not exposed to early, you'll never really, right? And
Yeah. Um,
horses are one of those
You'll
things.
be around cattle, but not horses.
Yeah,
That's
and
interesting.
I know, and I wasn't around cattle as a kid either,
Huh.
so I don't know why. There's something more, I don't know,
There's something
predictability of cattle
yeah,
behavior that
yeah.
isn't true of horse behavior, I think.
Yeah. Oh, yeah.
Maybe that's it.
I think people who are really comfortable with horses will still say, "Yeah, they're unpredictable."
Right.
So,
Yeah. Yeah, yeah. So, I really wanted to do food animal. So what are you going to do if you want to do food animal? Well, you could do you could do large animal internal medicine, but then you have to do, like, most programs are like at least half horses. Nowadays there are a
hmm,
few food animal sort of focused programs, but you still have to do horses for some period of time to do a residency in
I didn't know
internal
that.
medicine. Yep.
Hmm.
Same with surgery. And surgery was not my thing. Like, I was okay with some of the, sort of basic surgical, food animal surgical procedures, but orthopedics and stuff like that, no. Complicated things, no. I don't like being in a surgical suite. I can remember having a conversation when I was a senior vet student with a surgery resident, and sort of chit chatting, and I said, yeah, man, I really, this whole environment here, really, like, you got the gown and the gloves, and you can't do anything, you can't go anywhere, and you're stuck. And he goes, that's exactly what I love about it. So, there's something for everyone.
Yeah.
um
I think one of the first practices I worked at, we were doing surgery one day, and I was curious. I was thinking, I was like, could I do this one day? Could I only do this one
yeah,
day? Would I really enjoy it at the time that I start learning it? And I was like, and I knew that what is the saying that when you love doing something, time flies or something like
right.
that. And I asked her, I was like, um, do you just ever feel like time is flying whenever you're doing surgery? And she was like, no, that would be the worst. Like the dog's under anesthesia, like that would cost more money. It'd be more risk. And I was like, that wasn't what I was thinking at all. Um, and yeah, now my thoughts of surgery is like, it'd be really cool to enjoy it and have the time fly, but that like there's a risk of like, oh,
Right,
it's been an hour. Like,
right. Oh no.
yeah.
Yeah, yeah. Yeah, so I didn't want to do surgery. I didn't want, and there are a handful now of programs where you can do, like, in an academic setting, institutions, that teaching hospitals or whatever, that do practitioner boards, like food animal practitioner, dairy practitioner board, or whatever. There weren't really that many of those programs either, and if they were, I wasn't aware of them. And so I wasn't really sure what I was going to do, but I kind of felt like I wanted to stay in academia. Well, I had a DVM and a little bit of, I did an internship, but that's not, I needed something else. I needed some kind of credential.
More
And I was,
letters.
yeah, exactly. Theorio could have
Oh
been,
yeah.
I was really interested. And, like, even in vet school, I thought maybe I wanted to do Theorio.
I imagine there's even more equine stuff with that.
Yeah, but there are programs that are really focused on, like, cattle,
Okay.
um, and I'm not really sure why I didn't actually go with Theorio as I look back on it, because that could have been a really good choice. I don't know. Anyway, I was looking around for, what am I going to do, and I saw this ad for I'm looking for a graduate student. Um, the subject is, um, uh, whatever you're interested in, basically. Um, you'll do some field service, as part of your, what you get paid to do, and come do a PhD. And I was at Iowa State with Mike Apley, and I went up and kind of had an interview and talked about stuff and met some people in different departments. I met a couple of animal scientists because Mike said, well, you could, if you want to do nutrition, you want to do, um, something else production related, you could do something in the vet school. immunology, microbiology, whatever. He goes, or you could do pharmacology. I'm a pharmacologist. And here's how he came from, did a PhD in pharmacology, boarded in clinical pharmacology, and then worked in feedlot specialty practice for 4 years, and then came to academia to be a pharmacologist and feedlot expert. And um, I was like, what? Yeah, let's do that. So I didn't come at it from like, oh, pharmacology is interesting.
Mm-hmm.
I liked it when I was in vet school. Our pharmacology instruction was pretty pro forma, here's the drug, here's the mechanism, here's the next drug, here's the mechanism, memorize a bunch of stuff. It wasn't really clinical pharmacology. It was very much like, and nothing about some of the things that we talk about, nothing about evidence-based medicine, nothing about drug regulations or things like that. And so yeah, and I said, well, what kind of project am I to do? And said, well, I got some connections, so we'll find you something. And, found a company, sponsored a project. 3 projects end up being basically 3 chapters of my dissertation. So this new drug that was coming out, or that was being, in the process of generating um, data for approval. Um, what if we give the drug to healthy animals? Because the one of the comparator drugs, antibiotics, has been shown to maybe reduce inflammation in addition to killing bacteria. So, can we measure differences in neutrophils, for example, in animals that we give them the drug, they're healthy, we give them eat their drug, and then can we see changes in neutrophil function that might suggest that there is some anti-inflammatory effect or something like that. And then I worked with a microbiologist from the USDA, because in Ames, Iowa is the big agricultural research service. There's a diagnostic lab, the big NVSL, the big National
Mm-hmm.
Diagnostic Lab, and then there's a big agricultural research service, lab. And so microbiologist came over, and um, we he brought over the culture, and we kind of did this endoscopic method of inducing respiratory disease in a very controlled way. And then we induced respiratory disease. And the sort of novel thing that I did was, we had um, they look ridiculous now because of the electronics these days and the things that you can put into animals to transmit various kinds of health parameters. But they were like about egg sized radio transmitters that transmitted temperature.
Mm.
And we put them we induced respiratory disease in heifers, and we put these transmitters in vaginally, and measured temperature every 3 minutes from before we started inducing disease, and then followed them for they they came down with respiratory disease. They showed signs. We treated um, we had control group, and then 2 different treatment groups, the same drug that we used in the healthy animals, and the same 2 drugs, and then followed their temperatures over time, in addition to other clinical parameters. So that was the kind of sort of new um, investigation. And it was really interesting. And in a lot of ways, it was a kind of a small project, because it was a relatively short period of time. But the stuff that you have to do leading up to those kind of clinical, it's the same as if you do a pharmacokinetic study. Maybe you only take samples for 7 days, but the work you have to do ahead of time to get ready to do that project is, it's a lot, right? You've got to find the animals. Where are they going to come from? Are you going to buy them, or are you going to rent them? Where are you going to keep them? How many do you need? What samples are you going to collect? And then, because we were doing this GLP, or GCP, we had all kinds of paperwork, forms, SOPs we had to write, all that kind of stuff. So it was, it was months and months of getting ready to do this 10-day project, whatever it was. We did it at the Beef Center at Iowa State, and there was a little sort of hut out there that had electricity to it. So that's where he had the computer that um, that monitored all these radio transmitters, and I had to we had to tune each transmitter before we put them into the animals. I
Is
don't
that
even
like,
know what was going on there, but there was some sort of like process to basically get them ready to put into the animals.
mm,
And then I would go out there and monitor, make sure that all the equipment was working right. And I was sitting in the hut one day, one of the days of the study, and a helicopter went overhead, and the machine, I mean, just bananas, right? Because it interfered with all the radio transmitters. So we had a ton of data, every 3 minute temperature data. But we generated some cool data. The big thing I took away from that study was um, animals' temperatures go down after respiratory disease, whether or not they get treated. So even the control animals, their temperatures went up and then they went back down again after a few days.
hmm.
And also, we didn't show it. We didn't. Um, there was no statistically significant difference between the two antibiotics that we were looking at in terms of whether their temperatures went down faster or whatever. So to a different level. So that was kind of interesting. Sort of our hypothesis, but also interesting to sort of demonstrate that. Um, so I ended up with a PhD, and what I got out of my PhD and what my pretty sure my major professor wanted me to get out of my PhD was how to be a clinical pharmacologist, which is if you have a question you want to answer, who do you how do you pull together a team of people that can do the work to help you answer that question? So not necessarily that I didn't sit in the lab and do the neutrophil assays. I learned how they did them. I was involved as they were like writing their SOP and whatever. But I didn't sit in the lab and do all those techniques, and I didn't learn how to do all those techniques. But um, but I engaged with them. I asked them for, it's this collaboration kind of thing that we were talking about earlier, right? That you don't have to know all the things.
Mm-hmm.
But if you have the mindset of, I don't need to know all the things and I can put together a team of people and organize and manage all of those things and figure out how to um, ask and answer the questions I want to by pulling together all these different people, then that's valuable. So that's what I got out of my PhD. And also, as my husband calls it, the union card, right? Like a PhD is what gets you an academic,
Mm,
right? That's
yeah.
it matters less what your PhD was than that you have a PhD in
Mm-hmm.
some settings, right? Because that shows it shows that you have the level of expertise in something that you
Yeah.
can, and that you can pull together a whole thing, right? You can obviously I didn't do it all alone, and but I showed that, and if you have a PhD, you show that you can develop, plan, and develop and do a series of things and experiments, and then write them up
Mm-hmm.
and then defend them to a group of people because that's what a PhD is, right?
Yeah, you did the thing. You didn't just learn the thing, but
That's right.
you
You-
walked through the whole process.
That's right.
And whether or not something came out of that process isn't really always the goal. It's the fact that you now know how that
How
process works firsthand.
absolutely, yeah, that's absolutely right, and it's a really important point that if anybody ever asks me advice about grad school and or they're in the middle of a program and they're either where should I go or now I'm in this program and am I doing the right thing or whatever, your goal is not to cure cancer. Your goal is to get a PhD so that you can cure cancer, right? So when you're in grad school, your job, and this is the other thing in my household we always said was, what did you do today to finish your PhD? So every day you're thinking about, I mean, vet school is a little bit like that. It's just more regimented in vet school, not regimented. It's lockstep, right? You don't have to decide what you're going to do every day. They're going to
They
give you the stuff that you need to
Whether
do every
you
day.
like it or not,
That's right.
they have a plan. They're like,
That's
"You're
right.
gonna go to this class. Do I have to?" And it's like, "Yes, yes, you
Yeah, you
do.
kind of do. Yeah. Well, you don't, but
You can always leave. You just won't get your DVM."
that's right. So, but in a PhD, if you decide you don't want to, you don't follow through with the experiment or you don't write it up, which happens often it does happen some, right? Where, um, there's the apocryphal ABD, right? So, all but dissertation where you did the work, but you don't write it up. So it's
Hmm.
not science if you don't write it up, right? And you don't get credit for doing it. And so that's part of what is the value of the PhD is you did all those things. So I say PhD, it could be master's as well. Just usually a PhD is more, and often sort of broader. So I did three things that were all different from each other as the three chapters in my dissertation. So there were different investigations of, something along, they were all related to each other, but they required different study designs and experiments and things like that. So, yeah.
Here, real quick, ooh, bend this right here to like where it faces your here. Just there we go. It's probably not going to change anything, but
Okay. You feel
um,
better?
yeah.
Okay.
um, that's interesting that people will get all the way down the road and then not write it up.
Yeah.
It, I'm not gonna say it's the easiest part, but it sounds like you did all this work, and now all you got to do is write this paper,
Yeah.
which is a lot of stuff
It is.
to do. But it's kind of like the tree fell in the forest, and no one was around to hear it. Like,
Yeah.
what was the point?
Well,
What are
it's
we
fine.
doing?
Yeah. Yeah. I think a lot of things happen. I think you maybe burn out and you realize that you maybe were doing maybe you didn't actually really want a PhD.
Mm-hmm.
Maybe you just did that because that was the next thing to do, and then
Yeah.
now here you are and you do a lot of stuff and you really don't care that much about it. But there are scientists who do the work and then find it hard to actually write it up. Um, not and I don't know that it's so much about the like actual writing part of it, but it's sort of the you have to sit down and do this, then this, then this, then this. And so it's following all those steps. And then you have to submit it. And then you have to wait for peer review. And then,
Mm-hmm.
the first paper I submitted, I got back and it was, you know, in my view, it probably wasn't. I haven't ever looked back at it. But in my view, it was just like covered in red ink, right?
Yeah.
And I was devastated and I never went back to it. And so that happens lots of times where you just like it doesn't feel like it's worth the effort to try to figure out what you can do, how you can fix it, if there's some other, experiment you need to do. And so you just let it go. So
I mean, imagine that sucks a lot to put all your work and effort into this one thing, and then somebody looks at it in what half an hour goes, no, all of these things are wrong. You could have done this so much better. Or maybe it is, they don't even say that, but it feels that way.
that's right.
And then you get it back and you're like, okay.
Yes.
But I mean, right, I kind of mentioned earlier I was had a have a band background, at least in high school, for whatever that's worth. Because the professional music world is so much more intense, um, and a lot more on the line, being an adult and needing money. But the um the school I went to, it was like they would sit you down in an office. They're like, "Oh, wait, play the thing from beginning to end," and it's like every little mistake. It's not even just, "Oh, you played the wrong note here." It's like you played it the right note in the wrong way, either at the beginning or the end or at the very like 20 last 25% of it, or um this run doesn't sound right even though you played it right. And it's like, how about we change like every little thing? And you're like, what am I doing? Like, why? Like, I put all this work in, and then it's still never perfect.
No.
Like, perfect, it just never happens.
No, that's right. Yeah, and what leads that part of the world to be that way? Like why did they that's what's interesting to me is why are they like is that or
Hmm.
what service are they doing you?
In I guess in that example, because I still really like music. Like I'm kind of, I think I'm collecting instruments in my living room. I have now a keyboard and a guitar, and I have my trumpet from high school. Um, but no, what the band directors, at least when I was doing it, their goal is like to always be improving because you're either going forward or going backwards. You're never staying the same,
Mhm.
especially like because trumpet was what I was playing. The nature of the instrument, uh, if you don't play one day, you are going to get worse because of just how your mouth works. All those muscles just atrophy, which sucks. It's kind of the reason I don't play anymore because I'm learning the keyboard. Those notes are always there. Whether like my hands might be a little shaky and I don't really know that much about it yet, so I could just be naive. But yeah, if I picked up the trumpet now, it would not be the same as if I put it when I put it down years ago. Um,
It's like being an athlete, isn't it?
oh yeah, yeah, yeah,
I was not an athlete, but you were some kind of athlete, right? Where if
yeah.
you miss a day or you miss a practice or you
Mm-hmm.
miss a set, whatever,
Oh
then
yeah.
yeah,
I also played sports and it was definitely like that where you take a week off, which sounds nice,
but
but you come back and you're like 3 weeks behind.
uh,
Um, there are certain things, because like even nowadays I do I live in the kind of like physical exercise space. Like I love the gym. I recommend it to everybody. But there's kind of a concept where you can work really hard at something, and if you never take a break, then you kind of plateau, or you can start running into a wall that you don't see. And actually taking that week off can be beneficial. I think I heard some kind of timeline analogy for at least like building muscle, where this is like definitely gym bro mindset, but it was you can work really hard and you take a week off and it feels like you're like 3 months behind. But what you built in those 3 months will come back a lot faster since you kind of maintained it. So I guess going back to the music analogy, like I could pick up my trumpet. I wouldn't be as good as I was, but it wouldn't take me as long to get there
it's me.
nowadays.
Like if I picked it up, it would
Yeah, which like there's pros and cons, but
sure. Yeah.
um going back to the EBBM
Mm.
part of specializing, because I do want to specialize, and I don't know which specialties either in large animal or small animal require uh, uh, their own projects. Um, but since some do and some don't, I would, at least in my opinion, whatever that's worth, it it would seem beneficial for anybody saying that they're going to be a specialist in this specific part of the field, that they know or have firsthand experience in the process of building research, because everything we do as a doctor is built on those studies in some capacity. Like
Yep.
we learn in class all the time, like this thing does this, or it works like this, or blah blah blah, and we always say it like absolutes, when in reality it's like we figured this out slowly,
Yep.
and while rarely, but sometimes like foundations do shift slightly, um, but it's all built on that research, and especially like becoming a specialist like current day, um, you you walk out of a specialty residency and you're like, okay, now I am proficient in whatever's going on today. It's like that requires looking into the research that is most current and up to date, and then also being a part of it in some capacity. Um, I don't. I can imagine people would think going through that process like it could be seen as like a waste of time, but I don't know. I Like people say that about the EBBM part of the evidence-based veterinary medicine. For people who don't know, but the that part of the pharmacology, um, the a lot of people when we were going through it were like, "What are we doing? Like we're seeing papers about drugs we don't even know exist. Like we're not going back over these papers again. We're just kind of like practicing the skill and for what kind of thing." But no, I think it's important to some capacity, because I can't imagine sitting down with a client um, and your patient, and they're like, "Yeah, but doctor, why? Why is this happening?" And then you ask why enough, and you're like, "Okay, here's the paper." Begrudgingly, you can just hand it to them. It's like, "Here you go. This is why." Or the number of papers usually basing a good decision. Um, but yeah, you did you add the evidence-based veterinary medicine to the curriculum?
So when I joined the department and the first semester that I was really involved, it was a 2-semester course. I started in the summer. In the fall semester, I kind of basically sat through because there were other people teaching. And I don't even think I did any lectures. I might have helped out with labs because they were actual we had a 2 or 3 lab series with live animals, but most of them were, other things. We did at that time. The part of the reason it was 2 semesters was because he also did the anesthesia drugs, and so we anesthetized dogs. And so that there was a 2 or 3 lab, I can't remember now, series where we, you know, did physical exams on the dogs and did catheters and stuff, and then the next lab we anesthetized them and so on. But the in the second semester, the person who was doing a lot of the teaching at that time um, did an exercise that was related to um, looking at the evidence for um, was it antibiotics. There were monographs that a consensus group of individuals had created by pulling data from the literature. And the idea was that you would look at that monograph and follow the evidence back and say, like, do you see where this came from? Do you agree with it? That sort of thing. So it's kind of it was a different approach, but it was really thinking about, how do you what is the evidence? And so I I didn't use that exact thing, but the next year when I did it myself, well, me and another pharmacologist colleague, we taught both classes together. Um, we kind of came up with this. Well, let's be more intentional about um, how do you evaluate the evidence? How do you decide which evidence you need to evaluate? So that's the asking a clinical question. And then how do you find the evidence that you need? Well, that's the search part, right? And then, critical appraisal becomes an obvious part of that, right? Like, what is study design? What are the study design types? Which ones are most useful? How do you evaluate a paper to say this was a good study or this wasn't a good study? The reason you do that is because then you're more or less convinced about the effect estimate. So if it's a really good study design and it was big and all the things, then you say, well, I really trust this estimate of the treatment effect. And if it's not, or if it's a lower quality study design, then you say not. So I sort of gradually added things over the years. Um, I we recently put together, this is a paper recently published where about working with the librarians to do this, but I went back through and like from the first semester that I did things like what was it that we actually did, what were the assignments, what was the so I'm trying to remember exactly what that table looked like of the 2005 and 2006 is what we did, and then 2008 we did this and added things, and so sort of gradually built over time. And then um so we were doing it in the second semester of pharmacology, as I recall. Then we maybe moved it to, I don't know, it kind of moved around. But then when we did the big curriculum review and updated the curriculum and added courses and changed courses and removed some courses and downsized some courses, the that stuff we were doing that was EBV clearly was the practice of evidence-based veterinary medicine and how you do it and what are the steps and what are the skills. We sort of translated that into a public health course where you're already doing statistical methods and epidemiology and things like that. So there's sort of a logical fit with that course. So it became public health epidemiology and evidence-based medicine. Um, but the course was filled with um, already had the previous iteration of public health had a lot of assignments and was a very fully sort of formed course. And so then to add in this other stuff and then add more assignments and I was in, I was happy to still be involved. And so that's where we came with this sort of we'll do the contact hours in public health, but the assignments will be, in pharmacology. And then the focus of the evidence is about pharmacology. So um, so it's not um, all the other things that you can use evidence-based medicine for, right? Diagnostic tests of diagnostic accuracy and prognosis, and things like that. We sort of we don't do that because all the the assignments are all based in pharmacology. So um, and also because I was doing it, so it was going to be pharmacology,
Mhm.
right? So because that's what I'm most interested in. It's sort of the um, in some ways it's sort of a bit easier. Like the study designs are a little less complicated. They're not very statistical heavy most of the time. I mean, there's certainly statistical analyses involved, but they're often quite straightforward. So as a second year vet student, most of the time they're not super challenging to
Mhm.
sort of absorb as you're reading a paper, right?
Yeah.
It's not a lot of like logistic regression and logit models and like all those kinds of things. It's maybe you have a meta-analysis, but you sort of do a lot of blah, 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 as you read the meta-analysis and say, I trust what they did was good, and but here's what they end up with. So
It's exactly what I did.
yeah.
A lot of times they were like, we use this statistical method," and I was like, "Okay, cool."
Sure, you know what you're doing, right?
Yeah.
Yeah, yeah. And I mean, there's some easy. There's some straightforward ways to evaluate, right? So the kind of the flow charts that we use in the class, right, where you say, well, if it's this kind of data, and it's pretty most of the time you get a pretty good idea of is this categorical data versus continuous data, and you guys can assess that as a second year vet student. And so some really um, straightforward, I guess, principles of study design that are not for the statistician but for the user of the data, which is what,
Mhm.
like, for me, that's what I want you to take away is, as a user of this data, I understand where this is why I'm a proponent of at least some sort of research project. And I think a lot of residencies have some kind of research project. Maybe they're not doing a master's degree, but they do a project of some kind.
Mhm.
So you sort of see behind the scenes a little bit of how new knowledge develops. Um, but if you understand as a second year vet student the study design, basic principles of analysis, you can say, I'm going to use this data because I trust it because they explained it really well. Like that's actually probably one of the best um, ways to evaluate a study is, even though I don't really understand what you're doing, you told me what you did,
Mhm.
right? So that's, I think, part of the purpose of a lot of the we now have very good reporting guidelines for all different kinds of studies. And so if you do a really good job of reporting what you did, then even if someone is, maybe not a statistician or whatever, doesn't really understand how you use whatever the model is that you used, but you explained what you did, and
Mhm.
you explained why you did it that way, and you explained how you decided that it was the best way to do it, and you demonstrated that then all of that means you trust the results and feel like you can use that information, right, as a clinician to make a decision. And that's, to me, that's the point is, you're the user of the new knowledge,
Mhm.
right, as the clinician. And so you should be able to assess that new knowledge. It's like reading a food label, right? Like you don't have to be a dietitian,
Mhm.
but you understand what that label means, right? You know what protein is, and you know what those, you have a vague idea of what those, you probably have a really good idea what those numbers mean, but the average, you know, 25-year-old doesn't maybe doesn't know what all those numbers mean, but they have some idea, right?
Mhm.
Here's what I think. Here's how much protein I should probably eat in a day, and this thing has this much protein, so it's not a lot of protein. So, and whether or not that's whatever. So you're the user of that sort of end product of the analysis of the food product. So,
Yeah,
yeah.
because not everyone can be a statistician, and it takes a lot of time and effort to learn all those things and practice it, and then yeah, because as vet students and then going on veterinarians, especially in general practice, you can't be a absolute leader in every field that you are dipping your toe in. You can't be the greatest cardiologist, the greatest orthopedic surgeon, and the greatest ophthalmologist all within the same day, even though you're going to get those patients. Um, and you just have to kind of take it as you go, do the best you can. And I would say it works really well. We would know if it didn't. Like the whole, I guess, general practice system would fall apart if it wasn't working really well. Um,
Yeah, so to that point, one of the outcomes, right? A new graduate outcome or a competency that there is consensus is important that you as a student who has matriculates from a veterinary program that's accredited, um, is that you recognize your limitations and you do the common things well, and that which means that you are able to recognize when something is uncommon or is not responding the way you expect it to, and then you get help, you ask for help, you look in the literature, you refer, whatever it is. But you're not an ophthalmologist, but you are absolutely capable of looking at the 99 dogs that come in with the most common, the three most common or whatever it is things, and you're highly capable of doing that, and um, that is great. That is exactly what we want you to be. So yeah, that's a interesting thinking about the do I need to be a specialist in all the things? No, you don't. Um, and the some people say, the mixed animal practitioner is the most knowledgeable, right? Because they're seeing multiple species and multiple things, and but all of those things they're seeing are the common things most of
hmm
the time, and they get really good at those common things,
hmm
and then recognize when they need to, you know, read a book or read a paper or call a colleague or refer if that's an option. So,
I'm
yeah.
wondering because one of the podcasts I listen to, it's called the Checkup Podcast. I don't know if you
Mm.
know anything about. He's I think his name's Dr. Mike. Hope I'm getting that right. It's human medicine. But he talks about a number of interesting things. And at one point, they had a discussion about like the average person, not just medical professionals, looking at data and information. And I know the in that whole world, there's the CDC and then the FDA and all that, and then FDA for our world as well. Do you ever think that it would be beneficial for like the average pet owner or producer to be looking at studies or data like that? Because I know like it's complicated enough for people who live in that world and have the professional training. Um, like I think when I look at a paper, at least right now, I'm like, okay, if I can get half of this, um, like the introduction and conclusion, those I'm looking at those first, and it's like, what do they say? What happened? And then I'll kind of be like, okay, yeah, it sounds like that happened. Um, and like in for my personal perspective, like when I go back home and I talk to my family about it, like I'm hitting the basics. Whenever they have any questions or whatever, and it's like getting into that more complicated stuff, it would seem like a lot of work. But do you think it would be beneficial at all?
Yeah, that's a great question, and I think it depends on well, I'm thinking of who are you talking about in terms of the animal owner, producer, whatever. So, um, the the largest, most advanced animal production animal producers, animal production experts, um, people who run large production units, um, know the data as well as veterinarians do, right? Their system is extremely complicated. The margins are relatively thin, um, and they're producing a product at the bleeding edge, right?
Mm-hmm.
And so many of them are actually evaluating the original, the original papers, the data, whatever, or they are working closely with a veterinarian, a nutritionist, or whatever, who is sharing that information with them, talking through it, evaluating it, explaining it, or digging into it deeper or whatever. So, um, as compared to, an average pet owner.
Like the someone who's like maybe a good example is like what I've seen commonly is like young couple is like adopts a dog from a shelter. They're like, now I have a dog, and maybe they grew up with animals or they didn't, and they're like, you can see on their face like it's all new. They're like, what do I need to know? But they don't know the right questions to ask. Like,
Yep.
do you think that in that
Yeah, I mean,
situation?
to me, that's the beauty of the clinical veterinarian, right? Is you're the challenge, and also the fun part of is you're the translator,
Mm-hmm.
right? Um, it, you can't you can't push 4 years of veterinary training and 4 years of pre-vet, right, into a weekend of reading the literature about, right? So, um, because your understanding of your ability to read that paper, even if it's just the introduction and conclusion, is because of all the previous training you had, right? You know what those words mean.
Mm-hmm.
And not only do you know what they mean, but you know what the context of them is and why they're used in that particular way, and why they looked at this and not that, and whatever. And it's easy to forget what expertise you have, even though you don't feel like you have a lot of expertise.
Yeah, no, not at all right now.
And every vet student says that, but your expertise at this point is so much more than a person who hasn't had, 2 to 4 years of undergrad, and mostly in some either animal science or biomedical science or biology or whatever. And then add on to that, how many hours have you spent, even just in the first 2 years of veterinary school? How many contact hours? 30 hours, let's just say on average, 25 to 30 hours a week for 2 years. Well, for 18 months, right?
Mm-hmm.
9 months, 9 months, right?
Contact with like animals
No,
or
no,
just
you're
with the
like
material?
with the material, like in a classroom, in a laboratory, in a teaching setting,
Oh yeah,
right?
yeah.
And then add on to that your work in trying to assimilate all that and integrate it and apply it and take exams and whatever. And so if you think about like the volume of
Mm-hmm.
not just the like all the little facts that you know, but actually how they all fit together, because you
Mm-hmm.
do actually know a lot about how they all fit together, right? Because that's what you're supposed to be, right? Like that's what we're supposed to be teaching you, is how it all
Yeah.
fits together. You're learning the system, you're learning physiology, not what are all the organs called, right? You're learning anatomy, as in applied anatomy, and why do I need to know that versus
Mm-hmm.
that, and how does this fit together with that, and where do I find this thing, and and so the level of expertise that you have, even now in your veterinary training where you're at, is so much more than someone who is really excited about having a dog and wants to know everything and would read what you give them. Um, the, it would be a really big challenge for them to have enough information to be able to actually extract anything useful
Mm-hmm.
from the things that you, and that's so that to me is your part of your job, is being that translator to say, this is why this is important. Not just do this thing because I told you to, but
Mm-hmm.
here's why I'm telling you this, without all of the not necessarily with all of the everything but here's why I think this is true. There's some evidence that, not here's where the evidence came from, not here's how they decided, or how this is why I have confidence in this evidence. Just there's evidence that this thing, will be better than this other thing. And a cursory description of, well, in this one big study, and they randomized, and it was worked better in that study, but this other study wasn't as good. So, and that's in 10 minutes, you've said, uh, you know, 10 years worth of data, right?
Mm-hmm.
And 1,000 dogs. And so yeah, I think it would be, that's not to say that people shouldn't try to understand what's going on. I mean, when I go to my doctor and I want to, you know, tell me, why is that important, and what's the evidence for that? And of course I have a leg up, right? Because I know the lingo, but it's, human medicine is a whole another ball of
Yeah.
wax, right? And the words they use aren't always the same, and the diseases have different names, right? Because they're all named after somebody,
Mm-hmm.
um, and the conditions are, dogs don't get the same things as people, right? So
Mm-hmm.
they do get some of the same things, right? But we don't, you know, we don't look at cholesterol the same way in people as we do as dogs, right? Or the other way around. So anyway, um, yeah, to me, that's part of the beauty of your or the interesting part of being a clinician is that you're you're the liaison between this vast medical knowledge and this dog right here.
It would definitely be unreasonable to think that every or really any pet owner to like know as much as the veterinarian they're taking their animal to. Well, first, the veterinarian would become obsolete if that was true. Um,
So does that mean like if AI takes over, we will not have jobs? Like, what do you think about that?
I
What's
okay,
your current view on that?
my personal opinion, and
Mine
it
changes every day, so
well, mine's been somewhat similar. I'm amazed with what I can do. Actually, the most I've used it was for this podcast. Like I have it write a summary of the transcripts I create with it, and uh yeah, there's no way like I would have to sit down and like, okay, what did we talk about? Or I would have to go through back through the transcript, which is like over 100 pages long when actually written out.
yep.
And I'm like, nope, that would take way too much time. Um, so I think AI is a really cool tool, but it's just that it's a tool. When it first came onto the scene, what I was an undergrad when it was like first being talked about, um, or maybe even late high school. I don't know, but it was a big concern. And I have family members who are in the technology space, and so they know not only like the lingo and how things work. Because I'll be honest, when it comes to like the very specifics inside of how a computer works, I get lost. I learned out.
AI is kind of a black box, really,
Oh
even for
yeah.
the people that understand it,
Oh
right?
yeah.
Yeah, there's a lot of things where like, well, we put a bunch of stuff in there. We don't really know what's happening, but
Mm-hmm. Like I was talking to my older brother who knows a lot about computers, and he was like, "Yeah, it's just a bunch of matrices." And I was like, "What?" And he's like, "Yeah." And I was like, "What does that mean?" It's like, "We don't really know." But I was like, "Okay." I don't know how ChatGPT gets to the here's like a better way to write this, but cool. But no, and I think the um the common example that's used in our profession um is radiologists, how their primary job is or primary goal is to look at these images that these machines create and determine how the image looks, and then whether or not that's representative of the animal accurately, and if it is, what would that mean in that animal. um and to do that properly, right, you need to know how the machine works, which in our diagnostic imaging class is like the very beginning. We're like, how does this work? And it's like, we just scrape the surface of like physics. And everyone's like, okay, I trust you. And you move on.
yeah.
And I think we only talked about radiology specifically. So like x-rays, we didn't actually go into MRIs, which I think would have it would have yeah, my brain would have uh lost power. The but anyways, like that's been a concern. Will AI take over all the radiologist's job? And I'm like, I don't I don't think so. Cause what imagine that world. You would have a general practice veterinarian. You have an AI program either on their personal system, or they're using a system that is held somewhere else, and they put in that image and it spits out an answer, and you can maybe backtrack where the answer comes from, and maybe it can provide sources. But if it if you actually go back and you were like, okay, why did it give this answer, let me look at the actual sources it provided, even if it's 100% accurate, you're then putting in all this effort that while AI was meant to reduce time in the image reading process, it's now requiring more time from the people who um, don't have the time to do it. So, you can either trust what the machine is giving you, or then you need to verify what the machine is giving you, which isn't saving any time at all. So, maybe the the job will shift in the radiology space, and then you can expand that to every service within veterinary medicine. But I don't think it's going to take over anybody's jobs, because that's like technological advancement has always had that question. Is like this new thing comes on the scene and people are like, all these jobs are going to go away, and it's like, well, one, new jobs are probably going to pop up, and two, like, the world changes, you change with it. Like, that's how it works. And there are difficulties to it and a lot of empathy to be had, because um I know, like I mentioned, my my uh my family has some technology within it. They That's a weird way to say that. They work with technology. There's been a question of whether or not AI will take over programming, and in a way, like what I've heard, and I had a recent conversation this past weekend about it. It's like, yes, it's being used. It is not the best at it. It's really good at like really monotonous tasks that will take a lot of time. Just like the whole transcript example that I'm using it for. But at the end of the day, with the creating something that will solve a complex problem, or creating anything at all that doesn't need to be checked, will never really happen, because someone will need to observe the result, and there will always be a question mark on whether or not the result is either good or accurate unless someone checks it. And the only way to check it properly is to be knowledgeable and have experience in that field, whatever it is, programming, um, radiology. And yeah, so I think that's all to say. It'll be a cool tool. I mean, I've had fun with it. I think it'll be fun. I think it'll open doors. Um, Going back to the dog aging project, there's got to be a mountain of data that you guys have collected, and while it would be super cool to have just a team, like a whole building of statisticians, just crunching numbers all day long, if there's a really simple task that needs to be done, it's like go through all these numbers this specific way, to just type that in, upload the files, and then have that run, because it's very quick. It's like a really advanced calculator. You wouldn't be like, yeah, but it's really cool to draw out the numbers. It's like, at some point the calculator is here to stay. Learn how to use it, and we'll move on. Um, I don't know. What do you think about it?
I don't know. I uh, I have not engaged with it in the way that some colleagues have. Um, and mostly I just keep doing things the way I've done them, right? Rather than trying to, because it feels like it will take more time to figure out how to
Hmm.
use it appropriately or accurately or fairly or easily than to just
Sometimes,
do it myself, right? Do
yeah.
it the way I've always done it, right? So, but there at some point I will have to engage with it, and I would be doing my students a disservice if I don't acknowledge that. Okay, so the EBVM assignments, maybe it'd be better for I mean, at this point in the evolution of any of the AI tools, perhaps the assignment is you do it, and then you do, this is the AI tool, I want you to do the same thing. Have the AI tool evaluate the article and you evaluate the article, and your job is to compare them and see which one, and what's the difference between the two, and
Hmm. Whether or not you're correct or the AI is correct, and
or whatever. Yeah,
see.
and maybe not correct, but like what's different about it, and why do you think you got different answers, or why did you think
Hmm.
you got the same answer, or whatever. So it's an evaluation of the tool as opposed to just using the tool. Um, and that is a way to engage with the tools in a way that is um, involves critical thinking about it, right? Which I think is the the approaches that I've started to dabble some. I mean, or not dabble, read some, listen some, um, try to figure out how to use it tools appropriately in a useful way. Like I don't want to just do it to do it. So that's
Mm-hmm.
part of the reason I haven't really engaged that much. Is because I don't want to use ChatGPT or whatever or Notebook LM or whatever. Just to do it. I want to do it because there's a reason to do it. I want to do it because um, it's a tool that I think you might use as a student. Therefore, I should figure out how you're using it so that I can, so we can develop assignments or content or whatever to figure out whether it's a good tool or not. So I don't know. I'm not I'm usually a pretty early adapter on stuff, and this one I'm not. I haven't been. Um,
I've always been late to things. I've been pushing it off for so long because a lot of AI people, when I first saw people using it, it was usually out of laziness. And I have to write this essay. It can write it so much better than I can in like a second. So why do I need to sit here and write it? And then you have that battle between professor and student. It's like you didn't write it. It's like yes, but it's still good. Like it. So you had that example of having the AI evaluate a paper and then the student evaluate the paper and then comparing those results. Great in principle as a student. I definitely know people who are gonna just do the AI thing and then make up whatever comparison.
right.
Like whatever steps are on their end. Like there are people who are just not
Sure.
gonna do
I just
it.
want to get it done.
Yeah,
Yeah.
and
I'm not convinced that the learning I'm going to gain is worth it. Yeah. Yeah, I get that.
yeah, I because that's all that has now been a question with all assignments. Like, is AI going to affect the results that are being submitted by the student? And I spoke to one of the geneticists who puts on a study abroad trip to South Africa, Dr. Durr.
Mhm.
Um, this was a while ago. This is while we were on the trip. I did it in undergrad, and he had a lot of good perspectives about it. Where he was like, "This is a thing. It's here to stay. Like, it's not gonna just disappear whether you like it or not." And he's like, he was like actively trying to think of ways to have students not only be allowed to use it, but use it and then understand how it is used. And I think a good analogy is kind of like the calculator. While like the calculator quite literally is just a tool, I had some high school classes where they were like, yeah, um, we're gonna like either not allow you to use a calculator and then have all of these checks beforehand to make sure that you didn't have a calculator at all, when in reality you can't walk around in the world now without a calculator being nearby. Like, yeah, it's on your wrist, it's in your pocket. Like,
Yeah.
logically, there's a calculator around, so you can use it. So, um, or like there'll be there are some calculators now that I say now it's been since high school that this has been a thing that connect to the internet. And so like there's so many different tools and I don't know whether or not where to draw the line between okay, we gotta make sure you can do these things without the tool, but in reality, the tool will always be there.
Right.
Or for some tools, sometimes the tool isn't there. But with AI, I can't imagine a situation where it wouldn't be around in some capacity, unless it's like a the power grid has gone down
Well, that's
and
it, right? Like it requires power, right? So you don't have electrons, you don't have it. And that's
yeah.
certainly
But
there are
there
places where or times or events or
that affects a lot of things though, like in the
oh
hospital.
sure.
Like if the power were to go out,
Oh sure. Sure.
and you need to do everything,
Manually.
yeah, manually. Um, so I don't know. I
Yeah.
don't know.
The as a educator, I think about the um not so much the are you going to use the tool or not in your daily professional life, but my role as an educator is to demonstrate your competence and use an assessment that actually assesses your competence. And if I'm using an assessment that you can get somebody else to do it, which is what AI is basically, somebody else to do it, then that's my fault for giving you an assessment that so I need to figure out, and this is what it's happening across universities, is how can we what assessments can we use? An assessment means exam, assignment, project, whatever, lots of different things. How can we design assessments? Um, and what do we do, for God's sake, with online classes, right?
Mm.
How can we be assured that that there's an actual person, right,
Y-
doing those assignments?
Yeah, there's definitely a.
So, but that's a different that's a different conversation about which I know very little, because I don't teach online, like the truly online asynchronous sort of class. But um, I think, you know, the exams that we do in your program are um, they're not you can't use AI to do those, right? The assignments that we give you, unless we're doing them in class, and so that's what a lot of instructors, this is what I've been reading anyway, a lot of instructors, not in vet schools necessarily, but across universities, um, are moving assignments from homework to in class. So if you're a teacher of writing,
Mm,
students, if you send them home to do the work, some of them are going to use AI
yeah.
and turn it, and you might be able to tell, and you might not be able to tell if they use it or not. But you did. The fact is, you don't actually know. So now we're doing writing in class. You're evaluating it in the
Mm,
moment. You're using paper and pen, or a computer that I can see, a tablet that I can see what you're doing. Um, and a lot of them are using actual paper and pen. And because my obligation as an instructor is my, the state of Texas pays me to assure that when you graduate from our program, you have been assessed and determined to be competent in the ways that we say you should be competent. And if I don't consider how I'm assessing you and ensuring that the assessment is not that is of you and not another tool that you use that is not you, that's not engaging your brain in the same way, then that's my fault, right? Um, so the exams I have no problem with in my class, in our class, right? Exams are, but the assignments are, so how do I change them? How do I think about them? How do um, I don't know, evaluate the way they are currently working to make sure that, and not because it has nothing to do with like trust or integrity or whatever. It's my job is to assess whether you know how to critically appraise. And if you don't actually do the critical appraisal because you went home and,
mm-hmm,
dumped it in Notebook LM or whatever, then I'm not doing my job, right?
mm-hmm.
It's not, I mean, partly it's, I don't want you to, I don't want anybody to get credit for something that they didn't do. But that's, to me, that's a separate issue. It's certainly important. Academic integrity is absolutely important. But if my job is to determine or is to say, yes, you have the knowledge, and I haven't actually assessed whether you have the knowledge or not, because I can't tell whether that's you or not, that's that's on me to try to figure out how to do something different to make sure that, you know what I'm saying?
Yeah, that's an interesting perspective. I don't think I've heard that before, where it's the responsibility of the person teaching it to uh, determine that that student actually is competent in the thing. And with these new tools coming out, because there's AI is the thing now, there's going to be something next,
Sure.
whether in 2 or 10 or 50 years, and just to make sure that it the person who's essentially walking across the stage and receiving these accreditations that they are actually deserving of it.
Yep.
And because I've always heard it in perspective of the academic trust and did you actually do this thing? And if not, then that's really bad. Like, how dare you slap on the wrist kind of thing.
Right.
And I sometimes I'll have that mindset where it's like the like everyone needs to make sure that they're doing the thing, and if they're not doing the thing themselves and they're having these AI programs to do it for them, then it's laziness. They're being disingenuous, blah blah blah,
Right.
down the line. And in reality, they're just using these things, and it's available. And a lot of the time, like everyone I know who does it, like they could do it, and they are learning. But on some things, they're like, I don't have time for this, or I want to, do other things. So this monotonous work, like you do it for me, and then
Yep.
move on.
Yep. It's triaging your time, right?
Mm-hmm.
I mean, it's true of all students, but particularly professional students is you're uh they call them strategic learners, right? So what am I what and it's not deep or shallow. It's right. This is how much time I have in my day, and I need to prioritize my time. And so it's expedition, I suppose, or and also it's a little bit of the I I don't see the value in this. So that is also on me, right? As an instructor to help you see why it's valuable for you to do this thing this way instead of this way. If you do it this way, yes, it'll be done, but this is what you will miss. And if you do it this way, whatever it is, you're going to, you will get more out of it. You will be better at the thing that you want to do. So it's partly my responsibility to, I mean, it's just like my responsibility when we're talking about content, right? Is to say, um, this is important because I want you to learn this because this is something you're going to do. These drugs are the ones you're going to use every day when you're in practice. That's why you need to learn them. Some of it is inherently interesting, some of it is not at all inherently interesting. So it's my job, I say our job as instructors is to help you see why it's important that you know that thing. Even if it's just so you can pass boards, so you can, survive your fourth year, whatever it is. And maybe it's not so you can be a better, whatever, equine surgeon, but maybe it's so you can do other things that you care about. If I'm not engaging you in that way, then that's not all on you. It is also on me to make you see why it's important that you actually read the paper as opposed to have AI read the paper and summarize for you. Like I need to make it clear why that's important so that you say, okay, then I will do it that way because I know it's important because I know what I get out of it will be something I care about, not something that you care about, the instructor. So, right?
Yeah. Do you ever worry about the, I guess, it in hindering the actual ability of learning for the students? So, um, what I mean is an analogy of what I'm trying to get at is with social media, the kind of short form content has grown over the how many years, and subsequently attention spans are going down. Just colloquially, everyone's like, "Oh yeah, attention spans are the worst right now, and
Sure.
they're probably gonna go down even more." And for better or for worse, and I can imagine that with the use of AI in the classroom or outside of the classroom doing assignments or whatever, it can kind of offload the heavy lifting that comes with learning material. Um, do you see that? Because that is one of the downsides I can see for it. Do you know of any way to kind of get around that?
So that's a lot. What you just said there. um, I, I'm there's probably multiple trains of thought here. One is I don't think I maybe there is some evidence that some cohorts of individuals have some changes in attention span on a you know on a given day on a daily basis on a but I haven't and this is not my area, obviously, of expertise, but I don't know what the cognitive scientists are saying about, is it true that demonstrably our attention spans are changing, or is it just we're responding to what's in front of us, and if we went back to not, if we stopped looking at Instagram or whatever, we're fine. Like, we haven't
Mm-hmm.
lost that ability. I haven't seen any good data one way or the other on that.
Mm-hmm.
Um, Maybe there's some data coming out of like elementary school kind of kids that are on social media versus not or whatever. But I don't follow that literature enough, and I don't understand it well enough to speak on that. I don't think human brains are devolving.
Mm-hmm.
Like, that's not how evolution works, right? So I think our brains want to do something, and if our brains aren't learning because we're offloading it all to AI, they're going to do something else, and maybe that thing is really what they should be doing, right? So um, if uh, if I don't have to do long division because I have a calculator, then I can use my brain to do something else.
Mm-hmm.
So I think there's some of that as well. So because the human brain wants to, like, it, generally speaking, we are, if we didn't want to learn, we wouldn't be learners, and I mean, as a species, right? And we are learners as a species. So we'll just learn something else. If
Mm-hmm.
we don't have to use our brains to absorb some kind of content, we'll use it for something else. And maybe that something else will be something that we couldn't do before because our brains were too busy doing driving cars or whatever. And now we'll have the capacity to spend our brain power on something else. I don't know. That's the optimist, in me, as opposed to the kids these days, right? With no one has attention span anymore. Yeah, I don't know. I haven't seen. I'm not convinced that that is like a legitimate, actual change in our ability to attend to things. I think it's I think it's a challenge on any given in any given moment of our day to focus on the thing we should be focusing on, as opposed to, because I'm absolutely guilty of that. This is a lot more. Instagram is a lot more attractive than scientific paper, right?
Yeah.
On a especially at the end of the day, right? So does that mean my attention span isn't as good as it used to be, or is it just it's happens to be attracted by this thing that is, the shiny object of the moment?
Mm-hmm.
Um, so I don't know. And is it something I should worry about? Maybe, but that's like I should worry about. I can't worry about whether AI is going to take over the world because I don't have any control over that. So I got to control the things I can control, right? So
Mm-hmm.
here are the things I can control. I can control whether what I how I assess student learning, and I can, I don't know. So that's what I control my own consumption of media. If anybody asks me, I'll give them my opinion about things, but I can't control the world, right? So, and I'm not an elementary school teacher, thank goodness. I don't think I'd be very good at it, but I don't know. And I'm not a policymaker either, right? I mean, we can all influence policy, but that's such an sort of like it's such an end product of something that I don't think we can. There isn't policy that will directly affect that sort of attention span thing. It's about social media and availability, and should we have phones in school or not, and what do I allow in my classroom, or what do I expect in my classroom for attention, and so on.
Mm-hmm.
So um
Talk about something complicated. Is like how do you implement something on the macro to everybody that'll actually influence this one byproduct of the things that we're doing, um, which I don't know. That target's too. It's moving too much to
yeah,
actually do anything with.
exactly. And yeah, that's outside of my sphere in so many ways, right? So I got to focus on what I do have influence
Mm-hmm.
over, right?
Yeah.
Like second year vet students. Right? So yeah.
Because we're in the summer now, or we're just at the beginning of the summer. I imagine you're getting ready for the fall, for pharmacology, starting back up for the new second years. Um, as a professor, what kind of advice or anything do you have to say about learning in general? Because in in my mind, I think the biggest thing that I try and remind myself is if I can do something, I learn it better. So like hands-on activities will actually ingrain it into my brain a lot um, more effectively. And it's kind of hard to apply that to sit down classrooms, which is the first 3 years of vet school, for the majority, the vast majority of that time. And it kind of has to be because you're not going to, like, it would be irresponsible for the curriculum to be like on the first day, okay, here you go, and a pen with a bucking bull. Now figure out what's going on. And it's like that wouldn't work.
Right. Yeah. So your question is, how do you, how do you,
Like, do you have
how do I
any?
make that stuff more? How as a student can I make these things more concrete? How can I make my learning more efficient? How can I make it interesting? How can I not feel like I'm just rote?
Yeah, yeah. I guess on all of those or any of those of your choice, like, what's your biggest piece of advice?
For me, when I was in vet school, the things that I paid the most attention to were the things that I could connect to something that I already knew. That's why always in class, part of a classroom discussion is so think about or tell me about or do you remember or how many of you have ever seen, that's kind of triggering the knowledge that you already have so that you can connect it to this new thing that you're trying to it's sort of mental furniture that you already have and now you can hang or a connection or something that you already know that you can make a connection to. That is that to me that's key, particularly if you are not if you don't I mean there I was one of those people, but there are you have classmates, right, who like they can just sit down and memorize a list, right? And it's just a facility. And I'm not that way anymore, but I did used to be that way where I could memorize I can remember actually in pharmacology because those were the days of Kodachrome slides and overheads, and we didn't have note sets. You had a
Mhm.
notebook and you wrote notes. If you took notes at all, you wrote notes. So I've handwritten my handwritten notes. And when I would take a test, I'd read my notes. I don't have a photographic memory, but I would look at them often enough,
Mhm.
right, that I could see like, oh yeah, that was that in that line. So, but I don't remember that anymore, right? Because it's gone. What I remember is the thing that connected to the thing that I already knew, or the thing that um, the picture that I saw. I think this is where I think pharmacology. I think about some of the diagrams that Dr. Patterson uses that are to me really helpful. Like sort of you're visualizing here's the receptors and where they are
Mhm.
and therefore this is where so you already know and connecting the knowledge back to so remember in physiology when you learned about or that this is because in last semester in agents of disease you talked about streptococcus or whatever and where are those and where do they live and what kind of infections do they cause. Okay, so now let's talk about how we're going to what are some options for treating those? So it's the connections and it's the kind of integration of old knowledge and new knowledge and making your own whatever those connections are. I mean, some people love like mnemonics and things like that. Those are great for like learning content, but you're not going to do that when you're sitting in a exam room with a
Yeah.
client. You can't be like OOOTT, right? Like you
Yeah.
um you can't do that. So it's a great way to get started, but does that make sense? The kind of making the connections and as
Mhm.
opposed to like and this is where, students um who have a lot of experience in small animal and now we're talking about equine drugs. Like that's why it's hard for them, right? To assimilate that information. So how can we make that connection with, you know, mostly for me, it's what did you learn last year? And now I'm going to we're going to hang some more stuff on what you learned last year or what you're learning this semester in a different course. Um, so that's, that's to me, that's the big thing is if you can integrate stuff, if you can connect things. I feel like I learned that early in life and I don't know exactly when, but I've always kind of had that sort of how things sort of not randomly, but like make that's the connections that are, um, it's part of critical thinking is making those connections. So I think that's a big thing. And the other thing is retrieval practice. That's the
Mm-hmm.
key to learning, right? Is reading your notes over and over again and highlighting is not how you learn. You learn if you ask yourself, what is the answer to this question, right? Or someone else asks you, you quiz yourself, you quiz someone else. That is how you learn. And even if you don't, even if you didn't, if you don't think you know the answer, if you ask a question and you don't know the answer, and then you look at the answer, you will have actually learned it better than if you just looked at the answer in the first place. So that's the other key is retrieval practice. And but those it's the kind of cross referencing, especially in pharmacology. And I have found over the years that when I give students this advice, they do better. Like the what students that will come and see me and say, I feel like I'm not learning or I'm not performing the way I think I should. And is if they learn the content in kind of a very specific regimented way. Okay, I know this drug and I know these five things about it. And then I know this drug and I know these five things about it. But then if you're trying to say, well, what's the difference between this drug and that drug? You can't come up with that because
Yeah.
all you did was learn these things and learn things. So the kind of cross referencing, right? So what are what are the five drugs that have this adverse effect, right? How many drugs are associated with ototoxicity, right? And they're all different and but if you can do that, then you can make the other, the connections in other ways. And now everything about drugs is connected in multiple ways. So you don't, it's like going to the grocery store, right? Like if there's traffic, you're going to go this way. And if it's Saturday morning at 6 a.m., you're going to go this way. And if you only know how to go the one way, then, it's slower or you can't get there some days. And so I think about that when I think about like all those sort of what feel like disparate pieces of information about drugs is um, if you can get at them in multiple ways, then it'll be easier to access it when you need to access it. Whether that's on an exam or in an exam room, you'll be able to access it better.
Yeah, that makes sense. Because when I try and when I come across an exam question, I always feel best about my answer or the exam or the class in general if I can just walk through the process of getting to the answer. And honestly, I'll admit, pharmacology, I had a very, really hard time doing that because a lot of the times it was just me trying to remember the names because I was like, why is it named this? And it's like,
Stupid. Drugs are stupid. They all have stupid names. None of them mean anything. They're all spelled a bunch. And then there's new drugs every day, and so now like there's just a bunch of letters, and they
mm-hmm.
right? Yeah, yeah,
And and kind of going back to the clinical versus experimental pharmacology, it's like it does this thing, and then there's this like macro effect or these adverse effects, and they connect somewhere, but you could spend forever talking about that connection, and that's what a lot of physiology is. Um, what we learn with those two classes first year, but they can only go through so much, and I just that there's um a lot of gaps with where I was like, okay, it's called this and it does these things, and then you got to map that over the other drugs that do very similar things or work within the realm that you're discussing. So like all the cardiac drugs, and yeah, I remember I think it was the second exam, like the night before, I was like, I don't know anything. Like they're all here, but I don't know.
I can't
And
differentiate. Yeah, yeah,
so, but like I guess when it comes to names, like anatomy, I think does like anatomy in general as a field, you would say, the has a good system because they're like they have directions and then they have like uh foundational areas and they're like this thing
yeah,
is caudal and distal to this other thing and so it's called this
yeah.
and um I could always like figure that out and the name is like, okay, yeah, that name makes sense. In pharmacology, it was
No,
like
it
the
doesn't make
drug.
sense. It makes sense. I totally acknowledge that they don't. They don't, and it's yeah. I don't. And really, it keeps getting worse. Because I taught this semester. I taught undergrad, mostly BIMS majors, and most of them, at least 2/3 of them, are pre med or pre human, some human profession, health profession. And so we did. I spent a lot more time on human drugs, and oh my gosh.
Different world.
Oh man, the names of some of these, especially the new, like all the biologics, the kinase inhibitors and all the monoclonal antibodies, they all end in MAB, but there's all these letters in front of them that are all like Z's and C's and I's. I'm like, it's just, yeah, it's
It's like,
amazing.
where did this come from? It's like,
Yeah.
somebody just made it up.
Yeah, it's a medicinal chemist. That's who makes up most of those names. I mean, there are naming conventions, right? So, if a compound is in a particular class, like it does a particular thing, and it also has a certain chemistry, there are some naming conventions, like international naming conventions for that. But there's still like there's only so many letters, and so yeah, and then new drugs all the time. So yeah, it's pretty.
We talk about that kind of stuff in organic chemistry back in undergrad. Um, I could not pull that information to actually learn the name of it. Like, not once did you pull up the chemical formula of the drugs that we were learning. You're like, this is what it is. Like, because it's not the scope that we're focusing on.
No, and most of you are not chemists,
Yeah.
and most of you took chemistry because you had to, not because you thought it was really cool. I mean, there's always a couple of people, that are they're biochem majors or something, and yeah, and they were really into that stuff. But yeah, so there are some old school pharmacologists who think like, oh, structure will really help you learn and remember, and it doesn't. And I've never been convinced of that. So that's why, occasionally, there's and this is the sort of cross-referencing and differentiating, right? Is this is the maybe this is the third thing that's helpful for learning. So connections, retrieval practice, and uh, compare and contrast, right? Differentiate. These drugs are all NSAIDs. What are they? What makes them different from each other? Why do we use this one over that one? Why would we pick this drug over that drug? Can we use this drug in dogs or not? Things like that. So that's the sort of third thing that I think is useful. Maybe the cardiology drugs, you might like that be a way to, right? Like what's different about them?
Mm-hmm.
Um, how do you lump and split? Like, and that's a little bit of the cross reference sort of things, but how are they similar and how are they different? The compare and contrast helps you remember things. I think you did it. Didn't I give you a exercise like that, like with the antibiotics, right? Like here's two antibiotics. Name one thing that's the same and
Yeah.
one thing that's different, and that really helps sort of pull those things together. So, you asked me, or we were just talking about. I lost my train of thought.
All good. Talking about learning
Yep.
the the dumb convention of the drug names,
Oh
which
yeah.
isn't dumb. That's my word. It's there's a purpose to it, but it kind of goes back to the knowing everything or being a specialist in everything. It's like to know exactly why all the drug names are the way they are, and especially as they're rolling out like new drugs, you need to know all of the naming conventions within chemistry and how they're changing over time because those also change.
Right.
And
Is that knowledge that would help you be a better veterinarian? No.
I'm not interested at
No,
that
no,
at all.
I don't think it is, right? Like when I'm thinking about what do I think is important for you to learn, what do I think is important to teach, what is important to cover in a course,
Hmm.
um, is um, is what's the clinical relevance of any of these products? Are these um, and so anything that we look every year, like are we covering the drugs we should be covering? Are there drugs that aren't used anymore and we should stop talking about them? Um, are there new things that we should add? Are there, and so on and so on. And so the that clinical relevance is key to how we decide what to include, and then therefore how do we convince you that you should learn it, right?
Mm-hmm.
If it's not clinically relevant, well, it's historically relevant. Well, you know what? Most of those should not be in a course where I have 45 hours in a semester. That's 3 credit hours is 45 hours,
Mm-hmm.
give or take. What's the best use of that 45 hours to get you prepared for the next courses and for your first day in practice? Because that's first day, first week. That's what we're targeting, right? Not 10 years, not 5 years, like your first, because once you get out, then you start shaving stuff off that you're not using at all. You go into small animal practice, all that stuff about horses, not all of it. Most of it, goes away. You know, you go into general practice versus going into residency. You start shaving things off. And so what we taught you is now you're revising it under your own, making decisions about what's important and what's not. But so and where our job is not to teach you everything you need to know, everything you'll ever use, even on the first day. It's not. It can't be. We can't possibly do that.
Too
There's
many
not
things.
there are too many things. So we got to teach you the fundamentals and the why and the how. So this is really why EBVM is important to me, because it's teaching you the how um and the method so that you can figure out the how, right? So it's an approach. It's a like basic surgical approaches. We're not going to teach you how to do every surgery. You're not going to see every surgery that you'll ever do, but the basic principles of approaches, how to decide, what, selection of suture materials, how to hold your whatever, all those
Mm-hmm.
things, so that you could do it. And you also have the anatomy, right? So, and the physiology and the pathophysiology. So you could say, well, I've never actually taken that thing out before, but I know where it is. I know what it's composed of. I know how to do all the other things. So I can apply that and transfer that knowledge to this situation that I've never encountered before. Because that's what we're preparing you for, right? Is the thing you've never seen before. Right?
Yeah.
That's the goal, is to, because we can't prepare you for everything you'll ever see. You would be in school forever.
Yeah. No, I think that makes sense. That's a good approach, especially like 4 years is a long time, um, especially when starting vet school. But in the grand scheme of things, like it, uh, it's not that long. I wanted to say it was a drop in the bucket, but it's still a good amount of time. um, uh, but yeah, cool. Thanks for coming on. This is super fun. Do you have any final remarks? Anything for maybe students, maybe the average person listening, um, just anybody? Anything you want to say? You don't like these broad questions.
Uh,
Do you need some water?
it won't help. um, I mean, I always want to convince students that pharmacology is cool. And most of the time, if I can get them to think about why they're learning all the things they're learning and how they relate to whatever it is they think they're interested in, they realize that it's cool. So that's my advice, and it relates to, how do you learn? How do you be successful as a student, especially a professional student? Is um, Smart people, like vet students, can learn anything, but they won't remember everything if it's not related to something else that's important to them. And so, how do you have responsibility as a student. My job is to make it interesting, to maybe not make it interesting. My job is to help you understand why it's important for you to learn it, and your job is to like meet me halfway, right? And say, "Oh, I see that," or "I don't see that. Help me understand that," or figure out how on your own how to make it interesting or how to connect it with what you already know. So it's a I can't do all the work for you, and also you shouldn't have to do all the work, right? So it's we need it. Where we come together in the classroom to get accomplished what you want to get accomplished. So
Cool. Well said.
thank you.
Awesome.